Hello everybody, welcome to the Machine Learning for Drug Discovery workshop today. Nice to see you all in person here today coming to this event. We have a really packed agenda and really fortunate to have really great speakers and amazing, inspiring people today. So just wanted to give you a quick introduction to this workshop this year as a, for those of you that joined us the last 2 years, basically this year as a satellite event to ICLR, so basically independently organized and kindly sponsored by Helmholtz AI, who are funding, unfortunately, this venue here. So quite nice to have you around here. And just to give you a quick overlook over the agenda, we'll have a few sessions today in the first part of the workshop, where we'll be going through a number of presentations. The first one will be Smita Krishnaswamy from Yale University, who will be joining us actually via Teams, I believe, also from Europe. I don't know where exactly, but she's joining us remotely today. And then we'll have a networking session after the first 3 talks today, where we'll be kindly together just discussing some of the topics at hand. And then eventually we'll be going towards the evening sessions where we have, again, a number of really great speakers. We've had a short notice dropout. Debbie Marks unfortunately can't make it. We're still organizing a replacement speaker for her, but we'll be having, again, the 4 sessions from the afternoon docket as well. So without further ado, I already see that we have Smita around. So Matthew, if you could bring up maybe Smita's presentation to get started with. Smita, as you all probably know, is a leading researcher in, especially around biological investigations, especially around single-cell technologies, a lot of the tools that you might know and use coming out of her lab at Yale. And she's developing a really nice set of machine learning-driven discovery tools there. And she will tell us a little bit more about it throughout the session today. Do we have the slides? Hey, Smita.
Okay, I'm going to share my screen now. Let me know if you can hear me.
Yes, we can hear you fine. Thank you so much, Smita. I'm giving over to you, and we can see your slides fine. Thank you so much.
Is it okay? Can you hear me?
Yep, perfectly.
Okay. And you can see the slide moving, right? If you can, then I'll turn it off and then the next person can try.
She can't hear us. Smita, can you hear me?
Should I? You want me to start at 120, right? I can't hear you guys, so I don't know if anyone else is speaking. Okay, and so I can just get started now? Okay.
Yep, sorry. Technical
I thought I
problems.
was supposed to wait until 1:20, which is the time listed on the schedule, but that's fine. I can start. And let me know if you're having any problem. I'll keep looking at the chat because I have 2 screens. Hello everyone, thank you for inviting me to this Machine Learning for Drug Discovery workshop. Today I'll be talking to you about a series of works that we've been developing on generating and analyzing molecules of various sorts via graph-based signal processing and deep learning platform that we call learnable geometric scattering. So I'll go over the motivation for this, how that works, and how we've applied it to problems in molecular analysis and generation. In my lab in general, we are typically involved in high throughput, high dimensional data, of which molecular data was one sort. And we're wondering how to gain insights from this kind of data, which can often be unstructured or have irregular patterns of activity within it. And then fundamental challenges with every kind of high throughput data type we encounter, including molecular data types, is the heavy amount of noise in the data, but also the existence of a lower dimensional state space, which is important to learn for figuring out structure, patterns, multi-modes and multi-sample integration, as well as some kind of dynamics. And these kinds of problems we've now taken into the biochemical and molecular realm, and I'll show you a little bit about how we're dealing with it. So one of the main concepts we use is the idea of learning on a graph. Okay, graphs can capture geometric and topological information inherent to the problem. And they're very flexible objects. They can be used to model anything from molecular bonds to different kinds of interaction to, if you don't have a natural graph structure, you can use a similarity or a data affinity or a locality structure. And this can enable immediately, once you put things on a graph, techniques from both signal processing and deep learning to be used. And to be applied to fields where these kind of irregularly structured objects exist, such as biology and chemistry with the right inductive biases. An idea of what graph signal processing is, which is the viewpoint in which we think of how we're processing signals on molecular graphs. You can think of any features you have on nodes, or sometimes features you have also on edges, as signals on a graph. When you think about a signal on a graph, you're thinking about how this particular feature is spread and how it varies throughout the graph. And this enables you to use ideas from signal processing, such as low-pass filtering or bandpass filtering and these kinds of things. So the work, of course, for the flavor of signal processing that we've developed and we're doing is the graph diffusion operator. So the graph diffusion operator is just created from the adjacency matrix of the graph, which can be weighted. And you simply divide by the row sum of each of these to get a different stochastic matrix that gives you diffusion probabilities, starting from one vertex to all of the other vertices that you can transition to. And so this is computed as adjacency with the degree matrix divided out on the left. And we previously actually used it in the cellular realm, so this is kind of how we're taking insights from point cloud data, putting it on graphs, vice versa. And we use this kind of data diffusion operator to smooth and denoise signals on a cell-cell graph. And what it does actually in the frequency domain is if you raise this diffusion operator to the tth power, it gives you probabilities of t-step random walks, and you see the frequency spectrum of the graph, which is given to you by the eigenvalues, which decrease as you increase this t, and you see this pattern of low-pass filtering happening if you graph the eigen spectrum. And you see what's happening is that in the graph spectral space, you're performing this low-pass filter as if you're denoising the signal from audio or video. Another mathematical concept that we reach into is the manifold hypothesis. So we have millions of molecules that we're working with. We believe that the viable structures of them lie in or on low-dimensional or near low-dimensional manifolds or collection of manifolds. So that allows us to try to learn a lower dimensional representation that can cut away from some of the noise that it has. So in the molecular realm, we tend to do this with various sorts of auto-encoding frameworks. So this is kind of the recipe we use. We endow deep learning with geometry of this graph structure, topological properties, as well as signal processing in order to come up with predictions or generative tasks. And this is a recipe, again, that we've used in other realms. So with this, I can explain to you the learnable geometric scattering network. The original paper that first, where we first explained this came out in MLSP. First time we used something like that was in 2020, but very recently we have an IEEE transactions on signal processing paper that just came out that explains these transforms. And so the motivation, of course, is to learn biomolecular representations. So we want to learn representations of molecules that are faithful to structure as modeled by the graph. So if two molecules have, small, for example, graph edit distance, they should be close in this space. But we also want them to be smooth with respect to a meta property, such as binding affinity or energy, so that we can then predict these. If it's not smooth, it would be hard to predict these. And we want them to be invertible so that we can generate more molecules. So the molecules can be encoded as a graph. One way of doing it was with bonds. Another way of doing it is just with the 3D locations and taking an affinity matrix. Geometric deep learning has often been applied to this. So usually what people mean is graph neural networks, which translate operations that were successful on images, such as convolution and pooling two graphs. But graph neural networks typically work based on a couple of message passing operations. So the first is that every node usually gets messages from its local neighbors. And then it uses some kind of aggregation that's usually something like a mean aggregation. So the issues with this kind of message passing is it's fairly simple. What it's doing, first off, is smoothing. So it's always sort of low-pass filtering, and so that can lead to over-smoothing. And the fact that the messages are localized means it could also be underreaching if you don't do enough iterations, so you're like either over-smoothing or kind of underreaching. So to kind of show you why that could happen, you can take these two graphs, which as chemicals could be very different because there's this loop in this one and no loop in here. And you can aggregate these messages from these signals on to vertex 1 to compute its new features. But you'll see most of the vertices will have the same messages in both these graphs. And depending on how you aggregate them, you could actually lose distinguishability between these. Whereas if you look at graph diffusion, in one step of diffusion, for example, this message can reach out here. In two steps of diffusion, it can reach there. And whereas here, two steps of diffusion will still leave this node untouched. So different scales of diffusion with different powers of T can allow you to distinguish between graphs, and we tend to use differences between scales of diffusion, just like I'm showing here. And I'll get to that in a second, which is here. The differences in different scales of diffusion can be called as a diffusion wavelet. These were actually first defined a long time ago in 2006, but we started using them a little later. People in graph signal processing started using them, and we've been using them in learnable geometric scattering. We usually take differences in dyadic scales, and these are called these psi diffusion wavelets. So if you want to know what's different about diffusion wavelets, so if you just use one scale of diffusion, you would get this smoothing behavior all over again. It's just the message is being passed one way or another, everything is smoothed out. But when you look at these wavelets, they do capture different bands and frequencies of information that you can get on the graph, and that's what makes them a little bit more powerful. These kinds of wavelets were used in complex, deep networks that were actually fixed by Stefan Millat. And this at the time was called scattering transform, and people extended that to a graph or geometric scattering about 2018. So this geometric scattering transform usually consists of applying the wavelet to a signal and then performing a nonlinearity, which is usually an absolute value, but recently actually in AI stats just a couple of days ago we've shown that a ReLU can be a powerful one. And then you can build a cascade of these to create a deep operation, like a deep neural network. This is called a scattering path. But what we can do is we can actually learn the scales of these scattering paths to make it learnable. And then after you have node level representations of signals scattered from nodes, you could actually perform aggregation over the whole graph. You can use an MLP for the aggregation. Often what's used is a statistical moment aggregation where you take the moments of this signal that has been transformed by the scattering path that you see here. So our learnable geometric scattering took this kind of framework sort of generalized it. And we create a recurrent neural network diffusion module. We have a selector network for the scattering module, and an MLP for the aggregation module. So just a little bit more details on how that works is this is the diffusion module. It's a recurrent neural network that reapplies the diffusion operator to create all the scales. Then we select from specific scales here to get those differences to create the wavelets using a selector matrix. And each row of this should be softly one hot. So in a neural network, this is enforced by having softmax activation on each row, which gives you selection of filter scales. And thus the overall filter bank is learned, and you get a low pass as well as several selected diffusions, which give you band pass. And then you can get the aggregation module for which you could pick any nonlinearity. So for the alternating cascade path, we've shown that having two activations, a ReLU and a negative ReLU, are actually very useful, and they give you a very expressive bi-Lipschitz version of the scattering. I just presented that at AI Stats. And the whole construction kind of looks like this, where you could do this, and you can then get your scattering coefficients, and then redo the whole process, so that you can get multi-scale, multi-order scattering transforms. So we are able to show that we can relax the dyadic scales to any scales and maintain expressivity. And that's shown by the fact that if you have two different signals that, if you have a signal and it's given us increasing sequence of diffusion time scales with the wavelet bank, there's a lower bound on how much it'll squish the signal, so it'll keep the expressivity of the signal. So also there's an upper bound here, and what that shows is that it won't expand or blow up the signal so that it won't blow up noise. So in the restricted case of the integer scales we showed, we also had that lower bound which added injectivity and therefore expressivity to the neural network. And of course these operations are at the node level permutation equivariant, and at the graph level permutation invariant. So this reasons that we're not, this indicates that we're not necessarily reasoning about the labeling of individual nodes, but rather structures of the graph as a whole. So we've used this in a variety of deep neural networks, and I'll talk to you a little bit more about these. The first time we used this was in a geometric scattering autoencoder. And here we had a simple autoencoder structure where we used the scattering transform. And then we also had a regression here. And what this would do is it would select the scales of scattering that are important for predicting whatever property that is. And at that time, we again decoded the scattering via another coordinate neural network that was an autoencoder with an adjacency matrix in the beginning. But that would have some problems converging that we've recently solved. But what we would see in the embedded layer was actually very useful. We could see, for example, if we took a toy data set with graphs that are slowly changing, it would show a clean trajectory of graphs. So here, each dot is a graph. This is showing a collection of graphs that are all part of the trajectory, and this is our autoencoder is the one that's showing the right sort of structure for it. This was a bistable RNA, non-coding RNA molecule. And this non-coding RNA molecule is known to have two stable structures, and that is the visualization we get with the geometric scattering autoencoder, which we don't get with graph neural network and even other methods. So we started wanting to use this for molecular generation. So we've been working on that. We had a preliminary paper at MLSP a couple of years ago. We have sort of new results on synthesizing molecules for AMD. So we utilize the scattering transform of molecular graphs now together with attention over signals placed over atoms. And this attention mechanism allows us to generate better. So the architecture of geometric scattering bit synthesis, or GRASI, is the learnable geometric scattering, followed by an MLP encoder and a decoder. Also, we have a property prediction network. And finally, a way of generating that is actually a discriminator that decides that we've generated a valid molecule that pertains to certain constraints, and we found that the discriminator structure is very useful for enforcing these constraints so that the molecule, for example, the bonds are corrected and things like that. So we embedded all the molecules in Drug Bank, which is a database that you might be familiar with. Structurally similar molecules would be close by, and chemically similar molecules would also be close by, sometimes on another axis, because we've trained it to also be chemically organized by way of this prediction network. And then, so one of the things we can do is we can start with some molecule and we can optimize it to have better functionality while retaining its structure. And as we move in this latent space, we can generate out new molecules using latent space interpolation. And it can be latent space linear interpolation. So another way we do this is gradient ascent in the latent space. So that means we have that property predictor. So can we ascend that property? So we can do this by just using the gradients that we get from the regression network, and then we can optimize for properties such as molecular weight, solubility, and other properties of molecules. And then finally, what we have is an attention readout, which says which parts of the molecule are important for the properties we're seeing, so that chemists can look at it and see if this makes sense to them. And compared to other molecules, grassy generated molecules seem more valid, have high novelty, but still resemble the training data. And so this tells us that grassy with its structures is lying on the manifold with kind of the highest probability. It does have a lower Freshet ChemNet distance, which might mean that it's denoising some of the molecules out, so it's not generating some molecules in a noisy area. We've also used this basic construct here together with a transformer instead of geometric scattering when we only have access to the sequence. And this was published in Nature Machine Intelligence. So the basic idea here is that we can still learn a lower dimensional space if it's a sequence and not a structure using similar methods of attention. But the latent space we want to regularize in a similar way. So here again, the latent space is meant so that we can perform gradient ascent in the latent space. So we can start from a molecule and get to an optimized molecule. And if we've run it on different pre-existing start cases and shown that we get to molecules that are very similar to treatment level molecules, and we see that we get there much faster with gradient ascent. Here, basically what we do is instead of exploring the space locally, we can go towards the gradient of where a property is better, so that's what gives us the higher fitness path faster. And then once again, the attention tells us which parts of the molecule and specific residues are optimized or changing. The final method that I want to show is a method that's in preparation that we're hoping to submit soon, where we're studying actually proteins and their folding structure using similar techniques. This is, we call the proteascendant architecture. It's kind of a mixture of the grassy and the raster architecture. So we have a protein and we use both its sequence and structure here, and we perform the learnable scattering transform. And then we apply attention over the scattering coefficients. The scattering transform still has the scattering path and the aggregation. And in addition, now we have a positional encoding matrix for the scattering coefficients that we sum together with the scattering coefficients and feed to a multi-head attention network. And this multi-head attention network is then penalized basically for reconstructing the scattering coefficients. And we can often visualize the latent space and compute different properties, and also the 3D coordinates using this mean squared error reconstruction loss. So this is sort of an unsupervised network. And when we take a look at the latent space, you can segment the latent space, and the segments sometimes correspond to switching between different conformations of a single protein. And so what we're testing this on is on a coarse grain simulation by withholding data points and then having it interpolate or fill in back those data points. So when you look at the fate representation of another molecule, what you see is these distinct trajectories that are disjoint. And each of these disjoint pieces we see corresponds to distinct conformation of this protein. So we can do quite a lot of analysis by looking at the latent space, but then we can see what's going on in the real space by decoding out. And you might be wondering why we didn't need the adversarial generator here. It turns out that these attention coefficients have more information to reconstruct the whole protein conformations. And our preliminary results show promise in interpolating molecular dynamic trajectories, so we're hopeful that this could help speed up molecular dynamics trajectories. And we tested this with about 20% of the time points withheld. So just to summarize what I went through, Grassi employs learnable geometric scattering, and actually all our different networks employ the learnable geometric scattering transform. These form a multi-order, multi-scale wavelet transform that's done in a graph. So these are some very powerful operations that give us the structure of the graph. And we pair it together with different deep learning frameworks. And Grassi, what we did was we used an autoencoder setup to learn a good latent space, and then we used an adversarial network to generate out the molecule again. And now what we do is we don't often generate the initial molecule, we optimize it in latent space using gradient descent. We then copied the latent space techniques of this kind of thing, but together with a transformer, and we were able to repeat the same kind of pipeline with just using sequence rather than structure for protein design. And now we've been developing ProGSNN, which uses a dual attention mechanism applied to both residues in the protein sequence as well as the scattering coefficients to create temporally organized latent trajectories and identify changes in protein conformation through folding. And we're feeling hopeful that we'd be able to interpolate molecular dynamics trajectories using this. So most of this software, potentially barring the final features of the ProGSNN, are available on my GitHub if you want to play with it or if you have any questions on it. I just want to thank all of my collaborators. So the molecules that we're optimizing are for a disease called age-related macular degeneration, which people from my ophthalmology department are working on. And so it's their initial molecules that we're taking and improving. And these are some of my other collaborators on the mathematical and computational slide, and all the students and postdocs that are involved with it. And I'm happy to take any questions if we have a couple minutes time.
Very cool, Smita, thank you. Can you hear me all? Smita, can you hear me on the call?
But I can't see the room, so.
I'm sorry about that, but thank you so much for the great presentation. We do have some time planned for a Q&A session, so in case you already have a question, just raise your hand. I have a few questions prepared, but if you raise your hand, we can take that next. Smita, I had a quick question. So you raised a few interesting methods where you saw that there was a level of interpretability given through the attention mechanism. So I was curious if you ever tried to kind of interrogate that together with some domain specialists to see what the models found made any sense or was surprising in any way. Did you interrogate in any direction those models yet? Peter, we can't hear you, you're muted.
I'm not sure if you can hear us.
Right here in the chat. Some audio issues.
Oh, sorry, I can finally hear you now.
Okay.
Now, sorry.
Awesome. Did you hear my question before?
No, can you repeat it? I'm so sorry.
Okay, I'll repeat it for you. You interestingly showed us a few methods that had interpretability built in through the attention mechanisms. I was curious to see if you had interrogated by any chance with some domain experts, for example, some of the findings that these models highlighted, and if that made any sense, if that was surprising,
Do you have anything to share there qualitatively?
Yeah, actually, that kind of a little bit allows us to iterate. So I'm not a computational chemist, but we did work with some computational chemists, particularly in the AMD story, and they would tell us that some of the residues it was keeping track of and paying attention to would cause the molecule to be sort of sticky or slippery, and we were able to start at a different location which had less of that property. So we were able to add properties to train to our property prediction and choose starting positions using that. But I don't have the specific structures to release right now.
I had a quick follow-up, also based on what you just said. You mentioned in your latent space optimization work that it basically was reliant on some pre-trained properties that the model was utilizing for its optimization. So I'm wondering, have you also been able to kind of try something similar with an unsupervised approach, perhaps, where it's trying to optimize more generally unknown properties, for example? Is that possible in that framework? Have you investigated such a thing?
It would be difficult I think it would be difficult to optimize for unknown properties, but the basic setup is unsupervised. So we have largely the latent spaces formed by the geometric scattering and the structure that it captures. Within that structure, what direction you want to go, I think, is where the supervision comes in. And there's very specific things like you want the molecules to not be toxic, and those are named properties. But I'm not totally sure. But maybe if you train on several properties, then you get kind of a multitask boost, where it just becomes generally kind of a good and stable molecule. I have to formulate that a little more strictly before I can think about it.
Makes total sense. Any questions from the room? Anyone? A particular follow-up? If not, I have another one just about the validation of those methods. Have you yet tried, so you mentioned, for example, the generation task to be something that you're interested in. Have you actually tried to go back to the lab to test some of these ideas, and what are you finding in terms of the accuracy of your models in really reflecting what experimental findings would be?
Yeah, so the last set of molecules that I showed, we did put them through molecular docking software, and they seemed to dock to the right residues. So we've mostly been simulating them. Our next step is actually to go out and get them synthesized, but we're trying to do as many computational checks as possible before we actually go out and synthesize them. But generally speaking, we put them through, docking and other kinds of simulation software.
Makes total sense, and that was actually going to be one of my follow-up questions. I'm really curious in terms of the benchmarking that you apply here. Are there any benchmarks or metrics that you particularly like for these methods? What are the go-to things that you trust the most to really reflect strong performance of these models? Like, how do you interrogate this?
I actually, what I feel like people maybe should gravitate towards is not sort of these kind of canned benchmarks like uniqueness and validity, but maybe we should have more of these kind of benchmarks where you have molecules and you have certain properties and certain qualities you want in the molecule realistically. And usually what that turns out to be is not just is the molecule valid. It's usually a combination of properties that you want the molecule to have. And if we had, even if we went with drug bank and we quantified all these properties, another one really important one is immunogenicity. So these kinds of realistic properties, and people would benchmark how their method performs on them multifactorially. I think that would be kind of the most realistic kinds of benchmarks that we can have now.
That is super, super interesting. Last question from my side. I'm also interested in if one of the properties that you're able to optimize with your algorithms is also potentially around pharmacodynamics and kinetics. Do you feel like that's a problem that's approachable as well with your methods?
Are you asking me which specific properties that we're optimizing for? Some of the ones that I showed in one of my slides. So, in a lot of my research, the toxicity has to be low, immunogenicity has to be low. Sometimes it might have to be high, but other properties that I was showing, for example, in this slide are molecular weight, solubility for deliverability, number of saturated rings, and these kinds of properties.
Super, super exciting. Thank you so much. I think we all really appreciate the session today, and thank you so much for bringing this topic a little bit closer to the audience here. Thank you, Smita.
Thank you.
Appreciate it. With that, we're actually already ready for our second speaker today, and I believe that is the great Thor Grepo, who will be bringing us a little bit closer to the world of cellular reprogramming and the work that they're doing at Altus Labs. If you want, I think the slides are some of those machines.
Okay, no worries. Does this work? Can you hear me in the back as well? Because I'd rather avoid the microphone. You can hear me? Great. Thanks. Well, thank you very much for the kind invitation. I think this is so great, not only because of this particular event and idea, because it gave us an excuse to come to this wonderful city of Vienna. And I think it's one of the greatest cities on earth. And I hope you all will tap into that and make great use of it. I've termed this talk Beginner's Mind, in allusion to the Zen idea of having this open mind that you have to come to things with in order to fully grasp them, because I'm actually a beginner in this field, and I've really only been at it for a couple of years. Many colleagues, including Patrick, have been very helpful in onboarding me. But in addition to the ignorance that this brings, it brings also a certain open-mindedness and the ability to view things from a different perspective, and I hope I can bring that to you today. So I'm the global lead of computational sciences, AI, and machine learning at Altus Labs. Altus Labs is a relatively new biotech startup with the goal of using rejuvenation to create effective therapies. So here's the journey I would like to take you on today. First of all, something a little more personal, how I got into the space, if you like, and what AI, my old field, and rejuvenation, my new field, have in common. I'll then talk a little bit about what this means, what rejuvenation means, why I think we may have a shot at a new, maybe unifying paradigm in medicine. And finally, I want to talk a little bit about this dirty word, emergence, which is still one of my favorite words, because it describes all the fascinating things, all the surprising things. So let's start with this journey here. This is a Go board, and this is the game tree, or an illustration of the game tree of the game of Go. And this game has occupied me for many years, since I was a teenager. And a few years back at DeepMind, eventually we came up with algorithmic solutions, AlphaGo, to conquer this problem. And at the time, people thought it was a really tough problem. In any case, much tougher than the game of chess. But what I've humbly learned now is that the problem of drug development is so much harder. We were thinking about these combinatorial spaces here, but if I think about the multi-scale problem from molecule to organism that we're facing in medicine, in drug development, in biology more generally, this really pales here. And so this is an expression of humility and of appreciation that you all are dedicating your lives to making progress in this important field. Now, I immediately got into maybe one of the most ambitious, interesting, fascinating areas, I think, which is rejuvenation or longevity. Maybe it was a coincidence that I got into it because I got a call at some point with someone approaching me. But I just learned through this tweet from Carl Pfleger, by whom this list here is inspired, that in fact artificial intelligence and the field of rejuvenation and longevity have a lot in common conceptually. And I just wanted to point that out to you. If you think about it, both have a long history in fiction and art. And that really is testament to the fact that humans have been inspired by these ideas for a long time. Both how can we build an artificial intelligence, but also how could we address our mortality and live longer, healthier lives. And of course you see here Space Odyssey, one of my favorites, with AI Health 3000 playing a role here. And then this is the Fountain of Youth down here by Kranach. They're also both very difficult problems. Maybe some of the hardest problems that humanity has ever faced, but we have proofs of existence in both cases. We know for AI that the human brain somehow seems to be able to produce intelligence, a lump of matter, if you like. And of course, we also know that there are species, like the Greenland shark, who can get as old as 400 years. And we know that there are humans who have approached 120 years of age. So there really is hope, and biology shows us that these things can be done. Both topics are super important for humanity. If we make progress in them, they can do a lot of good. But there are, of course, also associated with both areas, AI and longevity, profound ethical problems that we need to consider. And interestingly, these problems have been so inspiring that there has also been a lot of cheating going on. Here's a little example, the famous chess automaton on the top right, where people already tried to fool others into thinking that an automaton could be intelligent. The fourth point here is that the underlying concepts of both intelligence and of longevity, aging, are difficult concepts to grasp. We don't quite know, or for a long time haven't known, where exactly we want to be going. But as soon as we define that more clearly, then I think we can make progress. Finally, and probably the participants here are testament to this, is that both fields are highly cross-disciplinary. And they have to, because they are spanning so many different problem spaces that really only when different specializations come together can we hope to make progress. So let's dive a little bit into this idea of rejuvenation. And to that end, I would like to show you the Altos mission statement, because that encapsulates our view on this. So we want to restore cell health and resilience through cell rejuvenation to reverse disease. That's basically the statement. So ultimately, we would like to reverse disease, and the means by which we want to do that is by rejuvenation. I'd like to explain this a little better. So if you take a look at this plot on the right-hand side here, you see here the age of humans, and here the deaths per 100,000 people, broken down into different disease areas by organs. And you can see here that as people age, these numbers go up in this plot linearly, so that because it's a log plot, this is exponential growth of the risk of dying from conditions in these different areas. So somehow there seems to be this underlying factor, age, that impacts all of these diseases. And now just imagine if we could somehow biologically shift the time back by 20 years, how that would impact the risk of disease across a vast set of different types of conditions. And so the idea of traditional medicine, of course, and of drug development as well, is to tackle each of these diseases separately. And of course, as you know, there are thousands of them. These are just really broad groupings here. And the idea that we're playing with is the idea that maybe we can tackle this one underlying cause and thereby develop therapies that are broader, that can cover more ground, where with one central principle we can treat many different diseases. And you know, if you like, we could call them foundational therapies. And just to show you the underlying idea in a different picture. So think of the black line here as the ability of an organism to withstand stress. And you know, as the stress levels go up, at some point these lines cross, and then we have disease. Now, for young people, when they're healthy, this ability to resist stress is still fairly high. Whereas when people get older, this ability goes down, and even smaller stress levels can lead to disease. And so the idea is then, can we somehow improve the ability of cells, of organs, of organisms to resist those stressors? And is there some maybe universal notion of resilience that we can strengthen so that we can tackle these different diseases all at once? Now, in some sense, we can liken this a little bit to the trend in AI towards foundation model. Remember, in the good old day, we had one data set, and we would train a model on it, and then that model would really only be able to tackle that particular data set. And then we moved through deep learning with the feature learning that made things more universally applicable. And now we're in the era of foundation models, where we build large models that can then be adapted to treat many different, to solve many different tasks once they've been adapted or fine-tuned to that. Could we imagine therapies, foundation therapies, that again come from this common core of strengthening organisms, giving resilience, and thereby can then be fine-tuned to address many different diseases? That's really the premise of our work at Autos. Now, when I was approached to join this endeavor, I had to think about, is this actually feasible? Is there any evidence that this might work? And since I was coming from the field of reinforcement learning, in particular applied to games, as you can see here, this is Pong. This is one of those old Atari game controllers. I don't know if anyone here has played with those old Atari game controllers. Not many. I think they died out. Anyway, so this is basically the setup for learning to play these games using reinforcement learning. But of course, the two key ingredients here are the upper and the lower box. We need to be able to make observations of the system in question, and there needs to be some kind of feedback system that tells us how well we're doing. Those are the rewards up there. And then we need the ability to act on the system. And really only when we have these two things in hand can we hope to control the system and have a purposeful effect on it. And so let me briefly go through what I think interesting reward models, if you like, are. This will be epigenetic clocks in similar systems, and what kind of actions there are that can actually affect rejuvenation, and gives us some indication that this might be possible. So, focus on that upper box here. And I would like to read you this, one of my favorite quotes by Lord Kelvin, which characterizes how important it is to put things into numbers. When you can measure what you're speaking about and express it in numbers, you know something about it. But when you cannot measure it, when you cannot express it in numbers, your knowledge is of a meager, unsatisfactory kind. It may be the beginning of knowledge, but you scarcely, in your thoughts, advance to the stage of science, whatever the matter may be. And so the critical question here is, of course, how can we quantify this resilience, this health, maybe this usefulness? And my colleague at Alto, Steve Horvath, has made some great progress of this with his epigenetic clocks. Let me walk you a little bit through his thinking. So he thinks of this quantity biological age almost as a latent variable that has various effects. You know, it has effects on the cellular changes and tissue pathology, on various diseases, memory functions, sarcopenia, frailty, and so on. And of course, it also has effect on things that we can directly measure on the molecular level. And he was wondering, you know, how can we quantify that? Because if we could then see what our interventions do and if they biologically rejuvenate. And so he formulated these axioms for biological age. He said, okay, it's going to be a quantitative measure, maybe also a Lord Kelvin term, which is highly correlated with age. That seems clear. It should apply to all mammalian species. We don't want something too narrow. You know, the biology is sufficiently similar with mammals that this should be the case. It should relate to mortality risk, but it must be different because it clearly cannot capture if someone gets bowled over by a bus. You know, it's somewhat different from mortality. And you would want it to relate to fundamental properties of biology. And in this case, the DNA molecule itself is the foundation of life. And so it turns out that the DNA molecule can have these methyl groups associated with it at the cytosines. And this DNA methylation plays an important role in gene regulation. And using a chip like this human EPIC chip, we can measure these little methylation bits along the DNA. Think of it like a huge binary vector. In this case, we can measure almost 1 million locations. And we can use that as input, because that turns out to vary with age, but also with certain aspects of just changes in health. And so what Steve built is a so-called pan-tissue epigenetic clock. And so the idea is that you can take different tissue types from humans, and you can measure that methylation vector for them. And then you do pretty much a regression, a penalized regression from that high-dimensional vector to the number you're interested in, health measures, mortality measures, age measures. And it turns out that when you do that, and these are all individual subjects here, and different parts, different tissues for them, that you can create a regressor that really captures age, where the predicted age is highly correlated with the actual age. And so we have a measure of biological age through the DNA methylation. So that's number one. So great, we can measure the effect and we know where we're going to go. But do we have any actions, any interventions available that actually do this well? And here we'll talk about the so-called Yamanaka factors as one example of such interventions. So in order to understand this, let's turn to, I think, one of the most beautiful and certainly most influential stem cells. But the fascinating thing is that during this process, there's also a process of rejuvenation associated with that, of epigenetic rejuvenation. And so effectively, the landscape is changed by a particular set of transcription factors so that the marble can roll back to its original point. And a lot of the work here is associated with my colleague Juan Carlos Belmonte. So there are these 4 transcription factors, OCT4, SOX2, KLF4, and CMYC, which can return somatic cells back to pluripotency. The problem is, if we applied that to a biological organism, that would be really bad because we need the cells to be differentiated to function in an organism. So how to overcome that? People found out, and you can read about it in this Ocampo 2016 paper, that you can devise a protocol by which you only, for a time, express those 4 Yamanaka factors. And there's a partial reprogramming going on. And then you stop it, and the cells find their cell identity again, but they are rejuvenated to some degree. And in particular, they tried this out on a mouse model of aging progeria, where they found longer lifespans, better skin quality, and so on, and also on older wild-type mice, where, for example, muscle regeneration was improved. Here are some more markers that can be measured in the progeria case, whereas in progeria cells you have this abnormal nuclear envelope, top left. Then if you have these rejuvenated cells, that develops normally. Similarly, in the progeria cells you have higher DNA damage, and that is lower if you apply these rejuvenation factors. And then there are these histone markers, in this case H3 K9ME3, which is also normally expressed in the rejuvenated case. And that is the one that compacts the chromatin into heterochromatin, so somehow is able to shut down and tightly regulate gene expression. Right, so I was convinced now. We had the observations and rewards, we have actions. Doesn't mean we have a solution, but at least we have, for these 2 important bits, some starting point of how we might be able to manipulate and measure things. Good. Let me now talk about emergence across scales in biology, because I also wanted to say something a little more general applying to drug development. And this is really the key idea here. If you think about what we need to do in order to develop drugs, we want to create an intervention at the molecular level. But then we want to have an effect on the health of an organism. How did he do that? Is there some way we can do this next? So, intervention at the molecular level, but health, the health outcome that we want is at the organism level. And so that means that somehow this intervention needs to go through all those levels of scale in biological systems. And they, of course, cross 9 or 10 orders of magnitude from the molecules to the organism. And ideally, as modelers, what we would like to be able to do is to predict what the effect of our intervention will be on the organism, and we would like to design interventions for a given target effect on the organism at the molecular level. Of course, there are other types of interventions. You can also intervene at the social level. In fact, one of the most effective measures ever taken against cancer was the banning of smoking in various places. So it's not the only way to do this, but I think at this workshop we're keen on injecting ourselves at the bottom level. So if we just look at 3 levels here, I think it's very important to understand that it's dangerous to only ever consider one level. For example, to consider just the tissue level. Why is that too narrow a view? Well, in some sense, there's stuff going on at this level, at the tissue level, and the level below tells us how that works, you know, how the constituents of the tissue come together to produce the tissue properties. And the level above tells us why we need to do certain things, the function of the tissue. And if you're familiar with Tinbergen's famous 4 questions about ontogeny, mechanism, phylogeny, and function, then of course it's exactly these questions of how, in this case, it coming from the mechanism and the why coming from the top, the function, the purpose. And so really maybe it's worth going back to how does this whole thing show up in the first place. And of course it is through emergence. We have physical laws acting down here, and as more of those come together and form higher level entities, stuff happens that's just not the sum of the parts that's happened down here. That means at each level, new, potentially surprising things happen, and we need different kinds of explanations that work at different levels of this hierarchy. And as I said at the beginning, I have more questions than answers, but I think this is a super important question. Because somehow, if you believe in this idea that we inject our intervention at the bottom scale and we look at effects at the top scale, we need to model through this emergent hierarchy. But all is not lost. We often think of emergence as being really tricky, but I would like to show you my favorite example of emergence. As I said, I'm a physicist by training, and I would like to show you the Rayleigh-Bénard convection as a phenomenon where you really see a fascinating pattern emerge just from the collective interaction of many particles under a thermodynamic potential. So what's happening here? I actually did this experiment at home. So you take a little petri dish and you put a viscous liquid into it, for example, vegetable oil. For visibility, I put in some graphite powder, which makes it extra visible what's happening here. And then you heat it from below, for example, in a water bath. And what you then see is that from the flat surface, we then see these so-called Bénard cells emerge in the liquid. And what you see here is that at the centers of these cells, hot liquid is flowing upwards. And then it cools down because it's cooler on top. And then that liquid goes outward of the cell and goes down at the edges of the cell again. And it's a stable phenomenon. This pattern can last for a very long time, as long as you maintain the thermodynamic gradient between the temperature at the bottom and the temperature at the top. The beautiful thing that I think gives a lot of hope is people have been able to characterize this quite precisely, because you can calculate this number here, the so-called Rayleigh number, from the difference in temperatures and other the viscosity of the liquid and some other quantities, and it turns out that exactly at the value of 1,708, you go from a normal diffusion process into this pattern formation. So we can understand these emergent processes and characterize them, and then potentially even control them. So you might be asking, what's happening here? Why is there some better explanation of why these cells emerge? And I think the most beautiful explanation that I've seen comes from, I think, a very general but completely underappreciated law, if you like, which Swenson called the law of maximum entropy production. Sounds a bit like the second law of thermodynamics, but it goes further. It says entropy is produced by a far from equilibrium system at the highest possible rate. So how does this work? As you increase the temperature difference, at first you don't see much in terms of emergence. There's just a diffusion process of the heat going from the bottom to the top slowly through diffusion. And then if you go here, suddenly the system has discovered a new mode of heat dissipation. And it does that because by not just moving heat as if this was a solid, but it now uses that it's a liquid. And you can actually transport heat by taking hot heat from the bottom and physically getting it up through that flow process to the top where it then cools down. And that's how these cells are formed. And you can characterize it and find that it actually maximizes the entropy production of the system, the rate of entropy production. And I think this is a beautiful image because if you think about it, that might just be what life optimizes for, entropy production. The sun is shining on our planet, life emerged, shines back as much heat as it can under the given chemical constraints which lead to the beauty of life. Good. Well, we're coming to the end, but I want to connect this with machine learning. And so here's another way of viewing this hierarchy, from molecular, the cellular, tissue, organs, and so on. And of course, it's not just a question of length scale, but it's also a question of time scale, because those things happen slower than those things here at the bottom. And now, fortunately, we have measurement modalities associated with this. We can do single-cell RNA-seq, bulk RNA-seq. We have X-ray crystallography, cryo-EM at the bottom. We have things we can measure at organism or organ level. And really, we have all of these tools at our disposal to characterize this emergent hierarchy. And I just want to end with a few questions. Namely, without understanding exactly what's happening, how it's happening, why it's happening at all of these different levels, can we build models that predict outcomes, health outcomes from interventions? Can we design for desired health outcomes the molecules we need to inject? And can we somehow model ourselves up this hierarchy, maybe in pairs, right? If we have, data for one level, cellular level, and tissue level, can we build a model that somehow mediates this and explains a bit of emergent difference that comes through this hierarchy? And then, of course, what kind of data do we need? Is it enough to have individual data sets, or do we need paired data sets that pair neighboring levels of this hierarchy? And can we then stitch a model together that goes all the way through? And finally, inspired by large language models and recent progress in foundation models, could it be possible that these effects of different biological scales emerge from training of simple data at the lower levels, just as we see new capabilities emerge in large language models? So let me wrap it up. I started out by telling you how AI and rejuvenation research have a lot in common and how I got into this. I then argued that there's maybe a possibility to finding a more unified approach to medicine by not looking at each disease individually, but looking at the resilience, the ability to resist stress for cells and trying to strengthen that. And my third point was also my first. I want to give you guys credits for being in the hardest game in town. It's really tough, I think, to do drug discovery in a principled way. But I think if we take this picture of the emergent hierarchy from molecule to organism and we can apply machine learning to these problems, then we can make progress. Thanks.
Thank you for this really brilliant tour through machine learning, regulation, and the parallels of which. I have a few questions, but they're really, really tricky questions. So, in case you want to save Tor from these questions, you could also raise some of ones that you have. Go ahead, Rob.
Thanks for the presentation. I get a little concerned with the use of English words like rejuvenation. And you mentioned, that's like you can measure things like the methylation patterns of the DNA molecule and all the omics of a single cell. What properties of those are correlated with what you care about in the sense of rejuvenation? Like why are we using that word?
Yeah, so the one pointer comes from the epigenetic clocks. So there are these epigenetic markers, and with age, we know that they change in a predictable way such that we can use them to predict the biological age of an organism. But also, we see that they vary with disease. So it turns out that if someone goes through a period of disease, for example, that then their biological age in that sense, in the epigenetic sense, accelerates. And then there are also interventions that reduce it. One of these interventions, for example, is the Yamanaka factor. So I agree with you. We need to be very precise in the types of words we use. This one suggests itself because of that connection, that we have actually established a connection between age and the epigenetic markers, and that we see interventions that move these markers in the way that it appears to make things younger again. So I use it in that sense, but I appreciate that we need to be very careful.
You touched on DNA methylation as one of the hormones of aging. There's also like telomere length and mitochondrial mutation aggregation. What are your thoughts about these like complex interplays of these different hallmarks? Are you going to focus on one particular, or do you want to tackle all of this in time?
Yeah, it's a great question. So in this sense, here we're just using the epigenetic markers in some sense as a correlative thing with aging. So, and it is correlated to a number of the hallmarks of aging, but not all. For example, not with typical. But overall, the way I think about it is that these are all interacting, and that we need to use a global approach to it. There's no fixing one bit because the other ones will come up. That's like the guacamole thing, you know, you fix one and the next one comes up. So I think we need these deep systemic approaches. And the Yamanaka factors seem to affect a number of them, at least, because you see organismal and therefore functional improvements, for example, in these mice. And that's always the ultimate measurement, right? We want functional improvements. Function as a modality, if you like, integrates a lot of the stuff happening underneath. And so if we can improve function, that's really the purpose. That's what we ultimately need to do. And of course, ultimately, to stay in this picture, function at the organism. Thanks.
Great question. We have another one in the first building.
Maybe just following up on that, you talk about the DNA methylation to age model as being something you can then use to kind of use in the context of sort of reinforcement learning. Clearly there you need like an interventional community, that you need a causal model, not a correlational model. Do you actually want to intervene and use that for?
Not really. So the intervention needs to be causal for sure.
Yeah.
But what you measure, not necessarily. Think of the video game.
Yeah.
The score
Yeah.
is not even part of the game, right? It's just some number that measures your success. So it needs it's enough to be one directional, so to speak. If the rejuvenated organism can be characterized by a lower number, say, of epigenetic age, then we're good. But it doesn't need to be the case
Yes, I hope so.
Welcome. Please go ahead.
Thanks for having me. Can you guys hear me all right?
Yep.
All right, and slides are all up and you can see them? All good with slides?
Yep.
Okay, perfect. I can't see anybody in the room. And feel free to also ask questions during the talk if you like. But maybe it's easier if you just take the questions at the end. And yeah, it's a great privilege to be here. Thank you, Patrick, and the organizers for inviting me to speak here. I'm going to tell you a bit about one tool that we recently developed called nascent, and then and how it can be potentially used in drug discovery in sometimes maybe surprising ways. And I lead a small team at Google DeepMind, and we are looking at using machine learning to better understand genetic code. So if you were to take the human genome and print it in books, it would be this full bookshelf looking like this. And the question is, what is how is the information about how to make an organism encoded in this genome? And one way in which we would know that we've done a good job of understanding this is if we were able to answer questions around how individual mutations affect different molecular phenotypes in the cell. And there are two kinds of mutations that we are looking at. So you can divide mutations roughly into coding mutations or variants and non-coding variants. Coding variants change the amino acid sequence of proteins and can therefore influence their function. And non-coding variants can affect the amount of proteins or RNAs that are going to get made at a specific time point in different tissues or cell types across the human body. And we've done some work across both of these types of variants. And on the non-coding side, Patrick mentioned the informer model. If you're working a lot with GWAS or non-coding variants, please go ahead and have a look at this method. And today I'm going to be talking about the left-hand side of nascent, so looking at how we can better understand genetic variants that affect the function of proteins. And generally, variant effect prediction, I would say it's a holy grail of genetics, because if we could do it well, then it means that we've done an important step towards of bridging the genotype to phenotype relationship, basically. And you'll see how, it seems very abstract and very fundamental, but you can still extract some useful knowledge out of these predictions, even for something very practical, such as drug discovery. In terms of how do we go about developing a system that predicts the pathogenicity of missense variants. So I joined DeepMind 4.5 years ago, and I was really excited because of AlphaFold. It was just a time when they were working on the second version of AlphaFold, and I was, really keen to see how we can also use it beyond structure prediction for genetic applications. So what is AlphaFold? AlphaFold is a neural network that takes as input the amino acid sequence of the protein along with multiple sequence alignments and predicts the 3D structure of the protein, and does so very, very accurately. As you can see here on the right, it's an example of an adhesin tip protein. And I think green is the ground truth and blue is the prediction, and you can see that they match almost perfectly, which is very impressive overall. And the question that we started asking is, what if we asked AlphaFold if we were to change this R to something else, a different letter, what would it do to the protein? And would it be able to, if it predicts a very different structure, would it be able to tell us something about the nature of this mutation that is probably pathogenic because it changes the structure a lot? And we tried this, and actually it doesn't work out so well. For one, because AlphaFold is quite insensitive to single letter changes because it's relying on multiple sequence alignment. So it's sort of extracting information across multiple sequences, not just a single sequence. And also, even if you are predicting the difference in structure, you don't know if this difference in structure is good or is okay or bad, because you ultimately don't have a mapping between the structure and the function of the protein, right? Which is what you would really want. So there's still like a big gap. Even if you could do structure prediction very actively, that would still be a question mark in terms of how would that affect, say, the fitness of an organism itself, because it opens up the whole question around the protein function, which is very, multifaceted and difficult. So we have to go back to the whiteboard and think about an alternative approach to this problem. So instead of using AlphaFold explicitly as predicting structures, we decided we're going to use it implicitly by leveraging it, internal representations and capabilities to help us with this problem. And if you look at AlphaFold carefully, you'll notice that it doesn't just predict the protein structure, but it also outputs other things. One of them is a language modeling head that does this, the BERT modeling task, where you blank out the amino acid in the input sequence, and you ask the model to fill in those blanks, basically, and you train this model using the standard classification objective. And if you actually use this output, and it has been in the field, we've been also inspired by a lot of great work from many different groups that have shown that protein language modeling or autoencoders on protein sequences gives you a very good prior on the distribution of sequences, evolutionary sequences, and it allows you to, and it's a very good proxy for predicting the pathogenicity of variants. So we thought that this may be a more productive route to start, and so we used the output of the language modeling output of AlphaFold. And the way you score a genetic variant is with this output head is you blank out the amino acid sequence, and then you compute the difference in loges between the reference amino acid and the alternative amino acid. And if this difference is large, this means that going from the reference to the alternative is going to make a big difference, and therefore the variant is potentially pathogenic. Now, this didn't yet reach state of the art, so instead we started thinking about how can we further improve it. And we leveraged ideas from the literature where people use a semi-supervised task where we have of variants that are frequent in the human and primate population. Because we know that variants that are frequent or in the human or primate population are likely benign because there are many people that carry them. So that's great. So we have a potentially a labeled data set, at least for one class, the zero class, if you like. But we still don't have data for the pathogenic case because everything that we have not observed in the human population, maybe because it's just that our effective population size is too small, so we didn't yet have the chance to generate this mutation essentially. Nevertheless, actually, it is, even though, but still, on average, I think it's more likely that the variants that we have not seen in the population are pathogenic. So we went ahead and we just used this set as our sort of positive set, as a pathogenic set, and then we further refined the language modeling head on this classification task. So predicting which variants are present in the population from those that are not present in the human population. And that turned out to work quite well, actually. So you can evaluate the system on different kinds of benchmarks, and there are two main classes of benchmarks that people like to use. One is looking at clinically relevant variants, the variants that have been classified as pathogenic or benign in the clinical settings, or mostly for rare disease. Or you can correlate your predictions with different experimental assays where people typically knock out the gene and then they introduce a mutated copy of that gene into the cell and then see if the cell still grows if present with a one-letter change. And the nice thing about these experimental assays is that you can measure every protein comprehensively, so you get like quite a nice complete picture for a given protein. But the challenge is that you can only measure it for a smaller number of proteins, and also the phenotype you're measuring in these experimental assays is a bit specific to the gene you're looking at. So the results may not be as comparable across genes, but within the same gene they're quite comparable. And we were very pleased that alpha-missense did the best on both of these benchmarks, because this is a very well-established field. Many methods have been developed, and it's quite a busy field actually. But many people then try to optimize their method only on one benchmark, such as Epstein-Barr. And doing so, maybe they inadvertently exploit some biases that were in the data set, which then causes the performance of this method to be worse, much worse on this other benchmark. So you're sort of overfit to a particular evaluation set in a sense, and that can be seen that the order of the rank of the alternative methods on these two benchmarks is often different, so then we were quite happy that alpha-missense did well across both of these benchmarks. And we looked into what the main drivers of performance were, and you see that you need to, fine-tuning on these variants helped a lot, and also having AlphaFold pre-trained helped a lot, and it was important to pre-train also on structural loss. And there are some other things that were quite important around uniformly, balanced sampling of the training set. So if you want to learn more, have a look at the paper. And then after we've been convinced that this is a good model, we decided to make predictions for all possible amino acid substitutions and missense variants in both the canonical gene isoforms as well as alternative isoforms in human. That then allows the community to basically use these predictions in different ways. And one alternative way to quantify the performance of your predictor is to choose your classification thresholds on pathogenic versus benign, such that your method is going to achieve a 90% precision or accuracy. And then you can see how many more variants is your model able to make a prediction that is above the threshold, basically. And you can see that if you compare E versus alpha-missense, you can see that there's a really nice boost in terms of how many variants more can you make a prediction for because of higher accuracy. So sometimes looking at these differences in performance can be a bit misleading because the benchmark may be saturating. So any small differences may be quite hard to achieve, but they might be significant on other more difficult benchmarks. One application of this is in rare genetic diseases. Here I'm showing you predictions for clinically actionable genes. The model does well on these kinds of genes, but I'm not going to spend time today talking about these use cases. Instead, I'm going to focus more about the potential use of these alpha-missense predictions in drug discovery. And I see three possible use cases for this in drug discovery, because after all, this is a machine learning for drug discovery workshop. First is you can use alpha-missense to flag important regions of the protein. You could use it to flag potentially toxic drug targets, and finally, you could use it to find more genes associated with diseases. And I'm going to tell you more about each one of these use cases, starting with the left one. So flagging functionally important regions, the way to do that is if you visualize your mutation matrix, where on the axis your mutation position, and your y-axis is the amino acid you're mutating, your amino acid position to, and then the color tells you the score, whether the model thinks it's pathogenic. This change has been pathogenic, shown in red, or blue, which says the model says this is probably a benign change. And you can imagine that functionally important regions of the protein are going to be mostly red, like flagging red because if you mutate, that you should not mess around with functionally important regions of the protein, because if you do, then this would decrease the fitness of the organism, right? And here's an example of a structured protein, which essentially hugs this other protein, and it binds in this sort of inner groove here, but it also has this binding motif here that is important for the binding of these two proteins. And here's the experimental data for this, and you can see that basically you roughly see these important sites as sort of vertical columns of red, and you can see that alpha-missense recapitulates them and also flags this particular binding motif as important, which is quite nice, because this means that if we didn't have this data available for this protein, we could have checked which regions are important, and then this could lead to new hypotheses. And for some proteins, like this one, alpha-missense did much better than some other tools like E. So you could benefit from more accurate predictions for those proteins. So that's one use case. It's basically telling you what region is important, and that might be important because it might tell you, oh, this protein is actually interacting with somebody else. If a region is important, is conserved, it has to be conserved for a good reason, right? So that might be just a hint to sort of pay attention to a specific region of the protein. Another application is flagging potentially toxic drug targets. Now, how do you do that typically? So when you develop drugs to target genes, to target specific proteins, you don't want to drug or target things that are essential for cell function, meaning that if you were to remove that protein, the cell will die. Okay? And there are two main ways in which people do that. Either they, for example, knock out the gene and see if the cells grow, or you can look at human genetic studies where the human population has observed a specific number of loss of function variants, and then you can compare whether you see a significantly lower amount of loss of function variants in the gene compared to what you would expect by chance. Because if you do expect a depletion of loss of function variants in the gene, this means that the gene is undergoing natural selection, right? So it is selecting against harboring these loss of function variants, and that tells you that this gene is probably conserved. Now, this statistical method called PLOF, or UELOF, is great, but the challenge is that if the genes are too short, then you lack statistical power to detect whether a depletion and the number of PLOF variants is significantly smaller than than expected. And this is roughly happens for 4,000 genes out of 20,000 genes, so roughly a quarter of genes is going to be, or a third is going to be underpowered with this statistical approach. In terms of essentiality for using experimental techniques, they're great, but sometimes they miss the right context or the right cell type in which this is performed. Right. The nice thing about genetics is that you have a full organism, right? Whereas if you perform experiments with cells in the dish, it's still cells in the dish, not the full organism. So there's a bit of a domain adaptation gap between these two. And the nice thing about alpha-missense actually is that if you average the pathogenicity scores per gene, it is a very good predictor of gene essentiality. So what you can see here is that for underpowered genes, shown in purple here, you can distinguish gene as cell essential versus cell non-essential as measured by some experimental assay much better than LUF or evolutionary conservation. For LUF, it's because these genes are too short and underpowered. For well-powered genes, meaning longer genes, LUF is going to do slightly better than alpha-missense, but they're still comparable. And that's really kind of interesting because, in fact, when we train alpha-missense, we try to balance. Between the number of pathogenic variants versus benign variants to be the same for every gene to try to, not not biased the model in any way, but that still seems to have come out of training, which is quite interesting. So yeah, this is, for example, one way in which you can use that. And a nice example of this in action is if you take the spliceosome complex. Like splicing is it's a very fundamental process in the cell of like cutting out introns to make the final mature RNA. And it has 7 subunits, and 3 subunits are very short, like roughly 100 amino acids in length. And for these, this Luft decile is actually not 0, which means that it's like roughly 50 or 30, which means that the Luft says that these genes are not cell essential, but it's because they're actually underpowered. Whereas if you look at alpha-missense, you see that all 7 genes in this complex are essential, which is what you'd expect because it's an essential component. So if you were to, say, drug one of these components, you could maybe disrupt their function in this spliceosome complex, and then the whole cell could not work functionally, and therefore the drug would be toxic, essentially. So that was an interesting sort of side output of alpha-missense that we didn't really expect, but it was quite interesting to see. And finally, how do you potentially find new, good drug targets? Obviously, drug target discovery is a very, very, very challenging field, and you have to get a lot of things right, and still keep your fingers crossed at the end that it's all going to work out once you go through clinical trials. But as the more information you can gather around from different evidences, the more confident you can be as you go through these. And it's been shown that drug targets that have genetic support are to that more likely to succeed in clinical trials. So it's a very strong piece of evidence that you get if your gene is associated with the phenotype. And there are 2 ways to find the gene from an association study from sequencing lots of people. So let's consider UK Biobank, which is I think currently it's the largest repository of genotype and phenotype paired data. And there are 2 kinds of approaches you can apply there. You can apply GWAS, where basically you're asking a very simple question many times, which is whether is a particular variant at one position, where you have rows are different people in the population, and you associate that with their phenotype, say whether they have a particular disease, say type 2 diabetes or not, and then you do a statistical test whether this association between these things is significant. And that's nice because it's very simple, and it's also it works quite well actually. It only works with common variants because you need enough variants to actually be able to have statistical power to detect differences. It only requires SNP arrays, which by definition measure common variants. But the challenge is, of course, that this doesn't return you the target of the gene. It only tells you that the variant is associated with the phenotype. It doesn't tell you which gene gets targeted, or whether this gene has been up or down regulated when having a G allele, for example. So there's a big interpretation gap that occurs after you've sort of analyzed your GWAS results. On the other hand, there's this, I would say, it's not a new technique, but I find that it's getting more and more popular now with the advent of more available data. Because the burden testing requires whole exome or whole genome sequencing, and it works with the rare variants. And the way it works is you have a gene, and in an individual, let's say you have these 20 variants or 10 variants, and then you have to construct something called a burden score, say between 0 and 1, to tell whether this individual in that gene, do they have a mutational burden, meaning that this gene is sort of possibly functioning less well. And then you can take this burden score and associate then that burden score with the phenotype. So you're not associating individual variants, right? You're associating the burden score of that gene with the phenotype. And that's nice because, first of all, you you know what gene you're looking at. Second, you know the direction, because higher burden score means it's going to negatively affect the function of the protein, so you know in what direction things work. The directionality of the effect, meaning that if a higher burden score is positive associated with the phenotype, then this means that you know that direction basically. Now the challenge is, of course, in this construction of this function that goes from a set of variants in the gene and the burden score. Like how do you get this function, right? So you're kind of asking that interpretation question sooner than later, as in GWAS. In GWAS, you kick the can down the road, you do your statistical analysis. Here, you upfront have to do this. You have to start thinking about how variants affect genes, basically. And one important category of variants that affect genes are obviously missense variants. I've been telling you about the missense variant predictor. So the question is how can you benefit this burden score? And one way to actually learn these burden scores is by using a neural network. So this is a technique developed in Julien Gagnier's and Oliver Stiglitz group called deep RVAT. I find it's a really neat technique where you don't hard code how you're going to aggregate the variants in the gene, but instead you're going to learn this from data. And the important thing to highlight here is that you can use many different predictors. So you can use many different features of variants. And one of the features that the authors have used was also alpha missense in this feature set. And what this method then does, so they use 34 different scores, and then what they do is they basically embed every single variant. Right, they push it through the neural network to get like a variant embedding, and then they do some aggregation across the genes. I think it's an addition operation, so it's like a set operation basically, and then they have a final module that then tells you the burden score, or they call it the gene impairment score. And obviously the question is, how much does having alpha missense help you in that process? And it turns out that it gives you a 10% boost in this specific setting. So if you include alpha missense, you find 10% and more gene trait associations than if you only used 33 different features. So I find that quite cool, because there's a lot of many of these predictors are still quite correlated, and they've already sort of exploited the simple features. So the fact that you can squeeze more out of the existing data, to me is quite exciting. I was very excited to see the results. And if you work in burden testing, I would recommend you to go and check out their method. I think they have a preprint on bioRxiv. And with that, I'm close to the summary. So I've shown you one of our recent tools, alpha missense, which is leveraging AlphaFold to predict the pathogenicity of missense variants. I've shown you 3 different potential use cases in drug development. So flagging which regions in the protein are important, which targets might be toxic potentially, as well as how do you use that to boost the number of gene disease associations when using alpha missense. And finally, alpha missense predictions are freely available. It's a CC BY license, and you can download them from Zenodo or Google Cloud Storage, but you can also access them through Ensemble's variant effect predictor tool if you're used to using that. With that, I wanted to thank all of my colleagues. It's been a real privilege to work with them. And also, I'd like to thank Julien and Eva for providing the deep RVAT slides and doing some of the analysis there. And with this, I'm very happy to take questions, and thanks for having me.
Very cool, Giga. Thank you so much for this exciting session. Again, we have a bit of a time for question and answers, so I'll just ask the room again if you have any questions. I have them here again, but maybe Rob, you want to kick this off?
Sure. Can you hear me okay? I was intrigued when you were saying that the first thing you tried was just looking at differences in the alpha fold prediction and using that as like your variant feature. And I was just wondering if you have an opinion, on why alpha missense does better than methods like Eve and ESM. Like, given that the AlphaFold delta wasn't enough on its own, but that's kind of like it's like a supervised pre-training thing. Like, but do you think that's the special sauce? Is that sort of intermediate step, or what's your take on that?
I think there's two things. One is that we're fine-tuning the model, refining the model. So like even language models, and Eve, for example, exploit the variability of sequences across the tree of life. But for some processes, for some genes, you maybe have to tweak this a bit to make the model work better. So, and I think that fine-tuning helps with that. The other thing, in terms of to your question, is like, if you can't explicitly use AlphaFold, let me rephrase your question. If you can't explicitly using AlphaFold for this task is not helpful, why would it be implicitly helpful, right? And the reason why, I think, is because the AlphaFold still has this very useful representation of where you are within the sequence. So language models, you don't know exactly whether you're in a disordered region, whether in a structural ordered region, right? So basically the structural context in which the variant is present is not very well present in language models, at least not explicitly. And when you use alpha missense, the model, because you're sort of mixing your representations with the structural representations, you're able to extract better context around where the variant is in the context of the structure. But that's different, right, than saying how does the structure change, right? So that's the key difference there.
And as a follow-up, given that the methods are so like the top performing methods on the ClinVar and the OMIM sets that you use, given that they're so different, have you looked at like the different error distributions of those and using those in conjunction?
That's a very good question. We have not. I think that would be really useful, like really interesting to perform like because the key question is like what are the different subsets to look into the different error distributions.
Right.
We've looked at only like ordered and disordered region a bit, but not systematically. So I think that's a very good good question to ask.
Thanks.
Very cool. We have another question.
Thank you for the talk. I'm interested in the pathogenicity scores. They seem a bit dissatisfying because they're a bit hard to interpret, right? How pathogenicity is actually acting and which diseases, which cell types. And also we have lots of variants that are kind of moderately pathogenic that probably don't substantially affect fitness. Do you have any thoughts about how we can try to deal with this and bring this information into these models?
One thing, the way we've approached this problem is that we've calibrated, so first of all, the model is fine-tuned to distinguish common from non-common in the human population. So there's the argument there that you're basically trying to calibrate your model in terms of fitness in the human population to some extent, right? Because it's really different, I feel like, because you may be asking different questions. We had a second round of calibration where we used variance in ClinVar. So really the way alpha missense is currently calibrated is you can interpret it as a probability that the variant is going to be pathogenic in ClinVar. That's how we've calibrated alpha missense. Now if you have a different set of variants that would represent something specific to you, right? That like maybe the probability, like a class of variants that have a specific property versus those that don't. You could recalibrate the method fairly easily on that task and see if that helps you. But yeah, I guess the short answer is you need to know what you calibrate against, right? Like you need to have the ground truth calibration set first. Recalibrating is easy. In terms of, I think your other question was variants that are potentially that don't have that are sort of deleterious, but they're not under selective pressure or something like this because they don't affect the fitness of the organism. Is that right?
Right. If you think about what Torres presented, a lot of these diseases affect people in old age, so they're important for humans.
Yeah.
And they're after reproductive age.
Yeah, correct. Yeah, exactly. So there's no selective pressure on these variants, right? So training on a little frequency is not going to pick these up. Because you have this inductive bias of training across genes, so you apply the same model across genes. You will learn maybe something in between, where you learn the deleteriousness of I think it's the question comes down to really mixing in also the function of genes. I think that's like if you decomposing the score into whether the variant is deleterious for the gene, so it affects the function of the gene, but does that then affect the fitness of the organism? And we have not decomposed these two things in alphamescence. They are sort of mushed together, and it's difficult to decompose them. I think. So I think it's a great question, but in my mind, we would probably need to decompose them more carefully to be able to decompose the two effects.
Makes sense, and with that, I think we have one more question in the room.
So when you were fine-tuning AlphaFold in this way, did you observe that the actual predicted structures also became more sensitive to these missense mutations or not?
No. So when we were fine-tuning AlphaFold, we were trying to make sure that the parameters of AlphaFold stay as close as possible to the original model. And also we were making sure that the model is still predicting the same structures as we were fine-tuning, so that the model did not forget suddenly how to predict structures. Because if you fine-tune these models, that neural networks can very quickly, happily forget things, just in order to fulfill the loss that you asked them, the task you asked them to do, basically. So we try to always make sure that the model was still predicting the same thing. So I would not expect any sort of fundamental shift in terms of how the model was predicting structures, and we were keeping track of the predicted quality, and that stayed roughly constant throughout. But it was quite important to keep training on the original structure. So I would say no. I don't think we saw any sort of change because we didn't want to see any change. In fact, on the structure prediction, because we went in with a different paradigm of tackling this problem.
Great, thank you so much, Giga, for this super incredibly exciting session, and as always a real pleasure hearing you speak. With that, we're moving to the next agenda item, which is a short networking and career break. If you are currently hiring, etc., please do make yourself known. I guess Novo guys are here. Altus, I think, probably has some openings. We, of course, at GSK, we have openings, so please look through the room and then just form some kind of networking. And we will be meeting back, I think, 3:50 for the next session, which will be Shantanu from the Broad Institute of MIT and Harvard. As always, we have a bit of time reserved for questions, so quick look for the audience. Any immediate... Sorry, go ahead.
Yeah, I was wondering how much more information is in the cell painting data as opposed to just bright field images? Is it possible that maybe somehow a lot of the information would be present in bright field images alone, thereby allowing us to scale even further at a lower price? Can't hear.
Are you muted, Chantal?
Sorry, I'm going to start again. Can you hear me now?
Yeah, okay.
Audible now? Yes. That's a great question. That was something I alluded to when I said there's a small revolution happening in virtual staining. And that's exactly what I meant, which is a lot of groups, I think back in 2018, 2019, in Cell Institute, folks in the Allen Cell Institute, folks in Google had shown that idea that you can sort of predict these fluorescent stains from bright field images. And ever since there's been a massive small revolution happening in the field in terms of people being able to do that. So I guess to answer the question, it is possible that for a large number of problems you want to solve, bright field may be sufficient. And I don't think we are there yet as a community. Some companies that have collected massive amounts of data are able to build these predictive models that can sort of almost replace cell painting in that sense. But I do think that is a very, very, very promising field. And there have been examples showing that it can work at least for a limited set of problems.
Very cool. There's another question in the room. Go ahead.
Yeah, thanks for the great talk. I wanted to ask in the opposite direction, like how much more information would you still say intuitively is in the full transcriptome, for example, or the protein expression? I saw you did some predictions of RNA expression already, but I was wondering if you have more intuition and if there's also like data to actually predict this. Is there a lot of cell profiler or cell painting data with associated transcriptomes, so you can test this and do these predictions?
Yes, absolutely. In the slides that you'll be able to download, there's one slide that is missing here, but it's there. You'll be able to download it. It's work by Marcia Hagegi in the lab, where she put together this really great resource of transcriptional profiling data and cell painting data that were sort of side by side. So it was a subset of which is something I showed you in one of the slides where we had L1000 and cell painting. So in that paper, we sort of talk about the complementarity and the shared subspace. So I guess the question broadly is, can we get more for these other assay types? And the answer is yes. The question that is asked is always going to be how much more and in what context. And that obviously depends on every experiment that you're doing. So keeping the costs in view, you have to make those trade-offs. But broadly, we've seen that there's definitely complementarity. Cost and the specific biology that you're exploring, it will really determine whether you need to do that. I'll also mention we have lots of new consortium now for specifically studying liver toxicity and figuring out if we can sort of augment or eventually replace animal testing for toxicity. And for studying that, we have off the bat taken a multi-assay approach. We're doing proteomics, transcription profiling, imaging across many different cell types, across many different physiological contexts. And that is exactly, the reason for that is because imaging in a single cell line, cell painting in a single cell line will not be sufficient to solve that problem, at least at the beginning. We might be able to get there. So there is complementarity for sure.
Makes total sense. Any more questions in the room? Otherwise, I have a quick one. Chantal, one thing that always interests me in this whole imaging domain is we never really look at the time dimension, especially when we think about dynamics being important for biologics. I'm wondering, when are we going to see like video type models that really understand the time dimensions of biology?
Yeah, I believe some groups are already exploring that. Your colleague Mindon in GSK is actually doing some of that himself. I think the constraint really, I don't think it comes down to the analysis. I think the analysis is going to be challenging. I don't think that is going to be the rate limiting step. The rate limiting step is always going to be the experimentation itself and producing time-lapse data. It's possible to do that. It's not trivial just because of, it thinks data can be more noisy, but it really is then a trade-off of scale and time, right? Time-lapse data is going to take more time to capture. So the challenge there is really going to be figuring out robotics and automation to make that data more scalably producible, rather than the analysis challenges itself, which will not be trivial, but I think the challenge really lies upstream in robotics.
Makes total sense, and lots to be still discovered, and I know you guys will be at the forefront of that. So thank you so much for giving us this session today, and really appreciate your insights today.
Thank you, Patrick.
With that, I can see we already have Lucy Kohler in the room. Lucy Kohler will be our next speaker in the second part of the workshop session. Lucy is based at the University of Cambridge and, as I believe, also part of Google DeepMind now, working on their protein and machine learning interaction models. And so I'm really, really pleased to welcome Lucy to the forum today. Lucy, are you already ready?
Yeah, hi, can you hear me?
Awesome. Go ahead.
Great, thank you so much. I'm really excited to be here. Thank you to the organizers for inviting me to give this talk. I'm also sorry that I'm not able to be there in person. I have a little baby who is just a couple of months old, and she's slightly under the weather, and so it seemed better not to make the trip. So I'm really glad to be able to present here virtually anyway, and I'm going to tell you about a bunch of work that I've been doing in collaboration with people both at Google and also at a couple of other companies where they've been kind enough to test some of the predictions that we've made. So we're really interested in using machine learning to predict protein function from sequence, and a number of our projects have therapeutic applications. And in particular, we've really had a lot of excitement working with this sort of active learning loop where we train and build models, use them to design sequences, and then test those sequences in the lab. And so I'll talk about a couple of projects to start with that involve that way of working. So of course, as we're all aware, there's huge amounts of protein sequence data, or sequence data in general, and this wealth of information keeps growing. And so the question of how to exploit this large amount of data has led to a lot of amazing work, some of which we've seen at this workshop and also more broadly. There's been enormous progress, I'd say transformational progress on protein structure prediction, and I'm actually interested in the question of whether we can make predictions directly from sequence of protein function. So sometimes even when you predict the structure of a protein, it doesn't tell you everything about the function. So I'm also particularly interested in how we can exploit this wealth of data to discover and design new proteins, and we'll touch on that a little bit later on in the talk. So I think there are two key challenges that we've been interested in, the first of which is protein optimization. So if you have a starting point, you want to be able to insert machine learning models in order to improve our ability to optimize a particular functional activity. And there's also protein discovery, where we're interested in discovering completely new functional activities. And I'm just going to turn the camera off so you're not distracted by my head bobbing around. But essentially, we're interested in training models that capture the relationship between protein sequence and functional properties or phenotype. It would be great to reduce data collection requirements and be able to transfer models and learnings between proteins so that we can really enable zero-shot design. And we've spent a bit of time trying to learn sequence representations where proximity encodes functional activity. This active learning loop has been particularly interesting for us, the idea that we can use experiments in the loop. Of course, this takes a fair amount of time and relies on our ability to find expert collaborators, but if we can use experiments to validate the models that we've trained so far, and also to tell us something about where those models are making mistakes, then we found that we've really been able to improve and fine-tune the models that we've trained. And so there are a couple of sort of interesting challenges in this loop, and in particular being able to optimize the model that we've made, so use that model to search the space and select the most informative next batch of sequences to test in the lab has been an interesting challenge. And I really like this idea of combining evolutionary and experimental data so that we can train models that relate sequence directly to function and sort of really combine those two wealths of information. In particular, the ability to screen large sets of exactly synthesized sequences, I think, is a particular strength of working with proteins as opposed to working with small molecules, where synthesis is much more of a bottleneck. So there are a few topics that I'll talk about today. First, I'll talk about protein optimization, and in particular, a few experimental collaborations where we've been able to show that machine learning can find highly diverse sequences with improved activity. And then I'll move on and talk about protein discovery. So we've focused quite a lot on model-based optimization, or MBO. This is the idea that we can use train a model, fit a model in silico on a relatively small amount of experimental data, and then use the power of compute in order to evaluate that model over really large spaces. We tend to adapt the model complexity to the amount of data that we have available. So we might start out training relatively simple models, and as we accrue more data, move on to training more complicated models. We typically try to take into account both optimizing or exploiting the model that we already have, and also exploring the space. So we set up acquisition functions in order to trade off between that uncertainty and exploiting the trained model. And then we're able to really, search the space in large scale, although, the space of sequences is massive, and so we're not able to brute force search everything. So we have to search in a sort of smart way, and then pick the set of sequences to test experimentally that kind of allows us to sample the space most effectively. And so the idea that we have a large batch size typically when we're running these experiments, and so we want to be able to use that batch size in order to explore lots of different parts of the space. And so we have a number of approaches we use to kind of prune the set of sequences and only test those that are quite different to each other. So we were fortunate to work on a nanobody optimization collaboration. We worked with two collaborators. Alpha Bio is a small company based in Seattle that has a very high throughput technology to measure nanobody antigen binding. And Lumen Bioscience is another small company based in Seattle, and they provided binding and neutralization experiments, allowing us to test some of the molecules that we found. And so we worked on identifying an antibody that had high affinity binding to SARS-CoV-2. We started from a nanobody that was known to bind well to SARS-CoV-1, but less well to SARS-CoV-2. It's called VH872. And we worked with about 46 of the residues in the sequence that were focused on the 3 CDRs and a handful of framework residues. And in each iteration, we were able to test between about 12 and 32,000 sequences. This was relatively high throughput. And then at the end, we were able to test a few nanobodies for binding using BLI and also for pseudovirus neutralization. So this project started a while ago, and some of the more common variants that emerged in the middle of the pandemic were not known at this point. And so we really lent on the OAS data set that the Charlotte Group at Oxford have put together. We used that to train initial models, and we use those models to search the space around VH872 and find, sort of put together an initial library that was between 2 and 4 mutations away from that starting point. In our first round, we then tested this set of sequences that we had used the VAE to help us find against SARS-CoV-1, SARS-CoV-2, and a number of other targets, in addition to 6 positive and 3 negative control targets, and we replicated each measurement 3 times. So this was a big sort of matrix of interactions we were able to test in one experiment. And we found that the data was fairly reproducible between replicas of that single experiment. So I'm showing that in that plot on the right-hand side of the screen. We also found that a lot of the sequences that we designed didn't bind, particularly when we got further away from VH872, 3 or 4 mutations away, something like half of the library wasn't binding. But we did have a reasonable number of binders, and we were able to use that as training data to kind of get our models started. So then in the second and third libraries, we trained both classification and regression models, We then optimized the predicted fitness in silico, and we selected batches of diverse optimized sequences that we then put forward for testing in the lab. So we were able to design both a second and a third library using this approach. And essentially, the classifier predicted, whether we think there will be binding. The regressor predicts if there is binding, how tight that binding might be. For the second round, we used an ensemble of light gradient boosted models. These are fairly straightforward models. We were doing multi-objective optimization across a number of different targets. And basically, we kept good sequences, but we really wanted to have a diverse batch to test. And so we clustered and selected the top sequences from each cluster. We also, in order to calibrate between rounds, included a lot of stratified sequences. And in round 3, we introduced an ensemble of CNNs in addition to the LGB models. These are all fairly simple models that we're training. We found that having different kinds of models basically improved the diversity of the design sequences. We also were quite stringent with our multiple hypothesis correction. We wanted to make sure that we were really seeing significant results or reporting significant results only. So essentially in rounds 2 and round 3, we had very, very many fewer non-binders. We were able to really increase the proportion of binders in the libraries. We were able to scoop in antibodies with significantly stronger binders in rounds 2 and 3. And we saw that optimizing the CDR loop 3 was really the best way to improve binding once we had that initial information. And so this is showing the strength of the interaction on the y-axis as a function of the distance from wild type, and we were particularly excited here that we were able to walk far in sequence space, so 15 mutations is the furthest we really went. It seems as if we could have gone farther. We were worried about how far we would be able to trust the models, but we were able to find really quite diverse sequences that retained or improved on the wild type binding activity against, in this case, SARS-CoV-2. And so we really discovered large numbers of antibodies or nanobodies that had binding activity. So we took everything that was significantly better than wild type and we called that a hit, and you can see that we had thousands of hits with binding against SARS-CoV-1 and SARS-CoV-2. Again, we found that these hits were really stratified across many mutational distances from that starting point. So we're seeing here that machine learning can really enable us to walk in sequence space in a way that perhaps we didn't expect, particularly because we're not using any kind of structural or sort of physical information here. We're literally just letting the models guide our choice or design of sequences. We looked at the diversity of sequences. We valued diverse candidates in order to ensure robustness against downstream constraints. And we found that we had a large number of candidates. I'm showing the top 100 sequences in round 3 on the right-hand side of this slide. And you can see that these are really quite a diverse set of molecules. We then chose 19 diverse sequences that were reported in the high-throughput assay to have strong binding and sent them for validation. Of those, 11 sequences had sufficient expression for us to run binding and neutralization assays. And as baselines, we included the wild type and the best single mutant sequence. And so this is showing binding affinities measured by ILA interferometry against SARS-CoV-1, SARS-CoV-2, and the Delta and Omicron variants. And these Delta and Omicron variants weren't included in our set of targets that we measured during when we were training the model. So this is measuring the ability of the model to extrapolate to new targets. And we can see that we have much improved binding against all of the targets for a number of the design sequences. We then carried out pseudovirus neutralization assays, and there again, we also saw that we were able to acquire the ability to neutralize Omicron from a starting point, which is shown in the bottom of these charts, which really had no neutralization activity against that particular variant. And this is showing the diversity of the sequences. The mutations were really confined to the CDRs, but there's a fairly diverse set of sequences that we found had this activity. So overall, we found that we're collecting really large amounts of training data there, but we are able to walk through sequence space simply guided by models trained on that experimental data. So I'm now going to move to a recent enzyme optimization project where we asked a similar question: can we walk through sequence space in order to optimize the activity of an enzyme, really just guided purely by high throughput data collection? And so here we worked with an enzyme called NucB. We wanted to improve its activity at therapeutic pH in order to potentially ask whether it could be used in wound healing. This is a non-specific endonuclease that can be used to degrade biofilms. And essentially the problem is that the pH drops by 80% at pH 7. The wild type pH is more like pH 9. And so we need to sort of get around this obstacle in order to be able to actually use this molecule. So here we worked with a small company called Triple Bar Bio, which is based on the West Coast. And they have a very high throughput microfluidics-based platform where they encapsulate variants of cells in droplets and then sort them based on fluorescence. And so they had developed an assay that related the activity of fluorescence measurement, and that enabled us to use this platform in order to identify variants that had high activity. So we can use this platform either to isolate top performers by simply sorting sequentially at higher and higher thresholds, or we can also use it to collect data for modeling, where we sort at multiple thresholds and then sequence everything that's in the pool, allowing us to construct a data set of sequences and accompanying labels. So in this campaign, we really tried to compare our machine learning guided models or campaign to both directed evolution and a very high throughput directed evolution campaign, and also to a hit recombination campaign. So we had a couple of baselines. We ran the directed evolution independently, but we ran the hit recombination in parallel with the machine learning. And we were able to carry out a total of 4 rounds for that comparison with hit recombination. And the directed evolution, we stopped after 3 rounds because we had quite an interesting performance comparison at that point. For validating hits, we had a kind of funnel of assays that went all the way from sorting very large libraries to testing whether variants could disrupt biofilm in a much lower throughput assay. So when we're comparing machine learning to directed evolution, the very high throughput bulk directed evolution assay, we found that after 3 rounds, we had significantly better activity from the machine learning guided directed evolution. And we were able to validate that top hit for biofilm content degradation, showing on the right-hand side of the slide. You can see there's quite significant improvement over the wild-type molecule. And we had 12x improvement from directed evolution versus 19x from machine learning. And machine learning was also much, much more diverse at that point. So it seemed like a interesting comparison. Here, we're now going to look at machine learning compared to hit recombination. So hit recombination we can track in much more detail. We can actually characterize round after round because the library size was smaller. And here, after 4 rounds, we looked here at hits, so variants that had increased activity over a single mutant, A73R, that already had 8x improved activity over wild type at neutral pH. And essentially what we can see is that using one of our machine learning based strategies, we're able to walk much further in sequence space, and we also have greater proportions of hits in that library. And here we do a simple clustering of the data, we can also see that we have much more diverse hits in the machine learning library than in the hit recombination. And so this sort of echoes what we saw in the nanobody project, which is that machine learning guided approaches can really explore a much larger volume of sequence space. And finally, I'm going to touch briefly on an AAV capsid design project that we worked on in collaboration with a couple of members of the Church Lab. So essentially the goal here was to design diverse capsids for AAV gene therapy. Goals in that field include preventing attack by the host immune system and delivering cargo to specific tissues or cell types. Our project focused on whether we could design diverse AAV capsids that were still able to package their genome. And I'm really just touching on this because we had a very similar outcome, which is that we can use machine learning guided evolution to walk much further in sequence space as opposed to the baseline, which is shown here in black and in grey. And we can retain activity much further away from the starting sequence. And this was published a few years ago. So this general idea that we can use machine learning to navigate through sequence space, potentially without including much information in the way of structure or in the sense of sort of physical information, this is something that I have found quite surprising, and I think has interesting potential for optimizing hits across many different modalities. So I now want to talk briefly about some work that we've done recently where we are annotating sequences, and I have a few different items here, and I'm going to talk about sort of all of these in a single swoop. Essentially, we wanted to be able to annotate proteins with functional activity more accurately than state-of-the-art methods. In general, we don't use alignment. We've found that pre-training across unlabeled sequences improves the performance of our models. We're particularly excited about large language models that we use to be able to translate between amino acid sequence and natural language. And I think the goal here is really to explore the space of natural sequences and even beyond natural sequences to find starting points which then can be optimized by the approaches that I talked about in the first part of the talk. So that's how these two parts of the talk really sort of fit together. And part of our work was inspired by this paper back in 2018 that really pointed out that many protein coding genes simply cannot be annotated with function. They focused on bacterial genomes. Things are perhaps even worse, if we look more broadly across archaea and animal kingdom. There's just like an enormous number of proteins that simply haven't been computationally annotated using existing approaches. And so this paper is a tour de force. They actually experimentally tested many sequences and were able to annotate them using the phenotypes that were recorded. But of course, the question is, can we use some of the advances in machine learning in order to do a better job of this in silico? And so we've worked on this for some time, and I'm going to talk about some early work that we did where we looked at using PFAM data in order to train models to classify protein sequences, and we've really built on that task in the years since. So PFAM is a large data set. PFAM-full is really grown using hidden Markov models. We focus on the seed part of PFAM, which is a much smaller data set of about 1.5 million protein sequences. A lot of the sequences in the different families are human verified, and members are chosen as kind of exemplars of families. So PFAM contains something like 17,000, 20,000 different families, and for each family there's a small number of sequences in the seed that are used to construct a hidden Markov model. So this is showing some information about the data from version 32. You can see that there's very many families with few sequences. So there's a lot of families here with a very small number of sequences, and then there's also a reasonable number of large families, and there's a very big length distribution between short and long sequences across these different families in this PFAM seed dataset. So initially we trained a very simple ResNet model in order to classify sequences, assign them to a particular family, and we built a cluster split of the data in order to provide a few short, difficult learning challenge. So here I'm showing the size of trained classes where there are test examples. You can see that again there's a lot of small classes, and they will have a few, 10 or 15 training sequences, and then a handful of test sequences. And we've split each of the seed families so that every test sequence is at most 25% identical to any trained sequence. So this is really asking the models to extrapolate in sequence space. And so we compared our models to BLAST and to a sort of HMM-based approach, and we found that our models, this is showing the performance as a function of distance in sequence space, where the left-hand side is the most distant, and you can see that the models are able to improve performance over those baselines. And what's particularly interesting is that the deep learning approach has provided complementary information, so if we ensemble between the HMM and our deep learning-based approach, then we get better performance than either of the approaches on their own. We use these models in order to add sequences to Vienna. We're able to increase the data set by annotating many more sequences, and we've since done this again, adding millions more sequences yet again. So what happens if we pre-train our models? Here we used a BERT style approach to pre-train models and then fine-tuned as a classifier. And so that was allowed us to improve performance a bit. This is the model now called prop-TNN, shown here in purple, which is improving performance over that ensemble of CNNs. And if we use a small ensemble of those transformers, then we can improve performance yet again. We then use those models to annotate something like 1.5 billion proteins that was released by MLDD as part of the magnified data set. And so one thing that's really nice about these models is they take a long time to train, but once they're trained, of course, we can use them to do inference on a very large scale. And as noted here, by the magnified data phase, we were able to annotate at least 200 million proteins that simply couldn't be annotated using traditional alignment-based methods. So more recently, we've been asking, can we go beyond this classification approach? Can we actually generate free text descriptions of proteins? And for that, we built a collaboration with the UniProt database. And we focus to start with on protein names, which are short free text descriptions of what a protein does. You can think about this as captioning. So of course, there's been a lot of work on image captioning. We want to be able to caption sequences. It's more difficult because a human can't look at the proposed caption and figure out if it's correct or not, whereas most humans can't. But fortunately, there are a number of highly skilled professional curators at UniProt who are willing to work with us in order to evaluate the performance of our models. And we worked initially with this T5 architecture, where we're basically asking the model to complete a number of different kinds of protein tasks. So we first lent on our PFAM cluster split task to show that, sort of validate the model, show that performance actually was even perhaps slightly better than the models that we had trained before. But this doesn't really ask the question of can they generate accurate free text descriptions. So this was using them for using the classification head of the model. And in particular, we noticed that something like 30% of the proteins in UniProt are uncharacterized. So they literally have the name uncharacterized proteins. There's about 250 or 230 million sequences in UniProt. So 30% of that is a large number. And so we were keen to figure out whether the models could make a contribution to naming these proteins. And so we took a set of characterized sequences and predicted names for those proteins. Of course, if our predicted names matched the existing UniProt name, then that was easy. We knew that the models were correct. But there were a handful of 43% of the sequences that we tested, and the names didn't match the existing name, and so we had to send them for curation. And so a good amount of manual corroboration was carried out. Unfortunately, 90% of that 43% still actually couldn't be verified. We couldn't say if the names were correct or not. But overall, 88% of the time, the curators found that the model meets or exceeds the semantic content of existing annotations. And we also had access to a model score, which we can interpret as a confidence, and we carried out some work to show that there was reasonable calibration of that score. And indeed, here you can see this is for characterized proteins on the left-hand side and uncharacterized proteins on the right-hand side. As the score increases, the ability of the curator to find evidence supporting the name really improves accordingly. And so ProtNLM at the moment has named something like 30 million proteins in UniProt. This number actually has gone down slightly, so we're continuing to make releases and to provide names to UniProt and to filter the names that we've provided. So this is sort of an ongoing collaboration. We've been working on this for a couple of years now. We made our first release back in 2022, and we've made a number of releases since then. Data are live in the UniProt database. We're always looking for feedback on those predictions. So if anybody has any names that we predicted they particularly don't like, please just let us know. Here's an example looking at some of those predictions in more detail. So more than 1,500 currently uncharacterized proteins were predicted to have a name that contains Cas9. To validate model predictions, we looked at the 100 top, and we used a workflow involving AlphaFold 2 plus fold-seek plus interval annotations to find evidence, or to try and find evidence supporting our predictions. The AlphaFold 2 predictions for 87 of the top 100 happen to be available in the AlphaFold database, and so we focused on those. 68 of the 87 were likely Cas9 homologs, so their top fold-seek hit was significant and was to a known Cas9 protein. 16 out of 87 were likely Cas12 or Cas13 homologs, and so only 3 were actually likely model errors. And you can see here a structural alignment between a known Cas9 protein and one of the uncharacterized proteins that's predicted to have that Cas9 in its name. We then asked whether we can query the model for a particular phrase. So here we've queried the model for the phrase small Cas9 homolog. And you can see as the prop LLM model score increases, that the size of the protein tends to decrease slightly. So it sort of said to us that perhaps the model's understood something about small. This particular phrase, small Cas9 homolog, actually isn't found anywhere in Uniprot. So we're asking the model to kind of interpret that phrase. And the blue stars out of the top 500 predictions, the blue stars show those where the AlphaFold 2 predicted structure returns a significant fold-seek match. So again, this sort of increases our confidence in the model's performance. So thank you very much for listening, everybody. We really found that models trained with length data are able to reliably discover functional sequences with improved activity. And our large models are able to annotate unseen sequences with free text names or captions that were found to be useful and reliable by professional curators. So if there are any questions, I'd be more than happy to take them now. Thanks again.
Amazing session, Lucy. I'm really blown away by what we're learning today. As always, we have a bit of a session for Q&A here, so in case somebody has a question already, otherwise, as always, Rob, go ahead.
Thanks for the presentation, Lucy. I really like the sort of like interface with the amino acid sequences, generating those captions, and it reminded me. I remember seeing some of that with that meta paper Galactica that was sort of like discredited, but they did some of that where they also looked at interactions between smile strings and proteins and things of that sort. Like, have you looked at that? What's next to come for interacting with these things via text?
Oh, yeah, I think that we're very interested in the idea of generating longer free-form functional descriptions that contain much more information than the short text descriptions that we've explored so far. We think there's enormously rich sources of information out there that we can use to kind of really increase what's known about proteins. So we very much view this as just a starting point. I think the idea of also looking at small molecules and sort of looking across those modalities is also really interesting, and that's something that I think we've been interested in looking at. We really encourage other people to look at too. There's sort of a huge wealth of tasks here, I think, that haven't seen much activity as of yet. Yeah, thanks.
I think we have another question here in the back. Should I come from? Yes, please.
Can you hear me from here?
Camera's on the
Yeah.
All right, cool. Yeah, thanks for the presentation. It's really cool. For the microfluidics and like other approach you showed, did you order like each specific sequence, or did you have like, kind of a library-based approach where because that's what people do often, right? It's because like academically it's cheaper. And if you order each sequence separately, what do you think of approaches to design more on a library basis versus explicit design of a single sequence?
Yeah, so I think this is a really important question. I believe that we actually ordered these sequences separately, but we used a fairly sort of constrained design region. I didn't talk about that. The full protein is slightly longer than we could manage with a 300 base pair oligo, and so we used a constrained design region. I think that the methods to make this cheaper and potentially more high throughput are really interesting and really important. And I think that I also would really like to move away from this modality in which you have to collect experimental training data for every assay that you want to make predictions for. So I think, I very much hope that, we've been collecting these data sets, but as we make them available to the community, people will figure out more and more how to transfer information between data sets and therefore kind of really reduce the amount of experimental measurements that are required, because it is really expensive, and also you of course can't develop these assays for every protein or functional activity that you're interested in. So I see that as a really important horizon.
Thanks.
Essentially, yeah. Thanks. Does that answer the question?
Yeah.
One more question.
Great, thank you.
Hi, great talk, thank you. Do you plan to expand your caption approach to full-blown descriptions, and then maybe also doing the reverse, as you have shown with the Cas9, smaller Cas9 homologs, that you provide a full caption, like really an abstract basically describing the protein and its function, to then get an amino acid sequence?
Yeah, I think that would be really nice, right? Being able to ask the models, query the models using English language for proteins of a particular property. I think that's something that we view very much as a challenge. Of course, experimentally validating that will be really important. Sort of looking maybe longer term, having the models be able to do all that optimization internally, again, sort of provides a way of perhaps moving beyond that requirement for extensive experimental data collection. So we sort of see this data collection as a necessary phase to go through on the way to being able to train models that can understand the shape of the relationship between sequence space and functional activity for different proteins and different phenotypes, and sort of navigate their way through it without having to collect experimental data in every case. Yeah.
Thank you.
Thanks.
Thanks so much, Lucy, for the really inspiring session today. Always a pleasure to hear you talk about this, and thank you so much for giving us this opportunity today to listen to you. Appreciate it.
Thanks.
With that, I'm pleased to announce the next speaker. As I told you earlier today, we unfortunately lost Debbie Marks due to her being sick today, but no less impressive, we have Pascal Monteur joining us today. You might know Pascal from his work including Eve, Eve-Former, Eve-Scape. I think Transception was also one of his papers. He's currently a postdoctoral researcher with Debbie Marks, working out of Boston. And he'll be giving us his talk today, just standing in for Debbie. I think we lost him just now. Is he still around? He'll be back, I hope, in a minute. Until then, let's see. He's back. Pascal, can you hear us?
Hey.
Awesome.
Thanks, Patrick, for the intro that I partially heard. Can you all hear me?
We can hear you, thank you.
Excellent. Yeah, sorry, Teams was asking me to reboot so I can share my screen. Let's try it now. Excellent. Good. So thank you for the invitation, and as you all figured out, I'm not Deborah Marks, but we've been working closely over the past few years on the presenting some of our joint work. So I'll be talking about language models for fitness prediction and design, and for those of you in the room who are not very familiar with protein design, there was a special issue of Nature Biotech a couple of months ago, focusing on protein engineering. And so if you're trying to get up to speed on protein design, I think that's a great a great place to get started. I highlighted a few of the papers that are either primers or review papers that can help you get started. I think with the fantastic talk from Lucy, I'll go through this very quickly, but what we What we are interested in with fitness prediction is to understand the fitness landscape, so mapping a representation of protein sequences and whether they are functional. And I think the critical aspect to keep in mind as you think about fitness landscapes is that there's not a single fitness landscape, but rather a plurality of landscapes that really depend on the experimental conditions. Like a particular protein might be functional at a given temperature and pH, but if you change those conditions, that protein may not be functional anymore. So that's something that is important in practice to bear in mind as for the rest of the presentation. So why might you care about fitness prediction? Well, there are like many applications of fitness prediction models, but just to highlight a few of them that I think are particularly important. The first one is human variant annotation. So there, the task is to help understand whether a particular genetic mutation in the human genome is going to lead to pathology. And so we, like experts over the years, have manually labeled a few of those variants. There's about like 2% of the human variants that are annotated, and the goal is to use those models to predict the 98% remaining mutations. Another important application is in viral escape prediction, and there the question that we are interested in is whether a given mutation to a virus is going to be maintaining fitness, right? So there's this question of whether it will escape immunity, but first and foremost, is that mutation going to preserve the function of the virus? And third, and I'll be focusing a lot on this in this talk, is in protein design. Whenever we want to engineer a new protein, well, we want those proteins to be functional in the conditions that we care about. So to train those different fitness prediction models, you have multiple data modalities that you may leverage. The three most important ones are sequence, structures, and functional labels. And they are very complementary, and each of them has pros and cons. So the sequence data, typically, that's the largest type of data set that you can leverage. Like now, in open, publicly available data sources, you have up to 10 billion sequences that you can leverage for your for training your language model. It's typically very useful in that it provides you with information about which sequences have been selected by natural evolution, and so from this you can get a good sense of functionality in general. But it can be complemented by other sources that provide more granular information. And one of such sources is structural information. So there you have more detail about the three-dimensional arrangement of your protein structure. Typically, you have like hundreds of thousands of different structures. If you have experimentally validated structures now with AlphaFold and other structure prediction tools, we have access to several orders of magnitude more of predicted structures that you could also leverage in your models. And that's a good complementary input to sequential information, and it's particularly very important when the function that you care about relies on interaction between pairs of residues, interactions between different proteins, for instance. And the last modality that is quite important is functional labels, so experimental properties that have been measured for for the different protein sequences that you care about. And there, I think the most important aspect of that data modality is that it really captures the notion of fitness in the environmental conditions that you care about. For instance, if you want to redesign a particular protein to focus, to operate in non-natural condition, very high temperature, very high pH. If you rely fully on what nature has sampled, you may be limited. Like if if there's no natural sequence of organisms that operate in those particular conditions. And so like it's very useful to leverage that third modality in that case. Those different data modalities have led to a wide flurry of different fitness prediction models, or like protein models in general, that you can broadly group into 3 buckets. There's the category of sequence-based models, where the goal is to learn a generative model of protein sequences only. Then you have the sequence label models, where you leverage both the sequence information and the experimental data. And lastly, structure-based models, and so that's typically other models to predict the structure, the 3-dimensional structure of a protein given its primary structure, so given the sequence of amino acids, or the reverse task of sampling sensible sequences of amino acids that may be folding into a particular 3-dimensional arrangement. In different generative models of structure, for example, in the case of RF diffusion, learning a good generative model of 3D folds of proteins. In the rest of the presentation, in the interest of time, I'm going to focus more on the left 2 buckets, but I will give some examples also of the right-hand side. So the structure for the rest of the presentation is, we'll deep dive into sequence-based models and sequence label models. But first and foremost, I wanted to tell you a bit more about how we may want to evaluate those different models with a benchmark that we put together that is called ProteinGym. And so if you're not familiar with ProteinGym, just to give you a quick summary of what it is, there are 4 key attributes of it. The first one of them is it's a compendium of different data sets. You have 2 types of data sets. You have deep visual scanning assays, where experimentalists have mutated a given wild-type protein sequence, operated all sorts of mutations, singles, multiple mutants, and experimentally measured the property of interest for that protein in an experimental setting. We also have clinical data sets where, on the other end, we have access to manually crafted or manually labeled annotations, like annotations from clinical geneticists, for instance, that have been able to say whether a particular human variant is pathogenic or benign, whether associated with disease or not. So, pretty large set of protein sequences for evaluation primarily. What we did is to implement a large set of models to our goal going in was to understand the strengths and the weaknesses of different models on a very broad set of protein families, to understand when an inverse folding model is going to be performing well versus a model that is typically trained on evolutionary sequences. And we looked at those questions both in the zero-shot setting and in the supervised setting. The other aspects of the benchmark are different performance metrics to compare those different models in different settings, and slicing and dicing those analysis to understand really the regimes where one type of model is performing better than the other one. And the goal was really to understand what works well in what condition to inform the development of the next generation of models. So I'll use this as a reference point throughout the rest of the discussion, but we found it to be a very useful tool to benchmark our own models and to understand what works well and what does not. So perhaps you will find it useful as well. So let me talk about sequence-based models a little bit. I'll start with the the traditional way to go about it was to first extract a multiple sequence alignment, so a set of homologous protein sequences to the protein that you care about that are all aligned in the same coordinate system, and then to train a generative model on that multiple sequence alignment to learn the distribution of amino acids at each position. And like those models, historically they were focusing on single residues at a time, so ignoring dependencies between residues, and then progressively they've baked in more dependencies between residues, so looking at pairs of interactions in POTS models, for instance, or looking at potential interactions across the full sequence with, for instance, variational autoencoder-based models that is represented on this slide here. So they work very well, surprisingly well, I would say. But like they have a few limitations, and one of them is that because of the fixed coordinate system, they are not typically able to score or handle the insertions and deletion mutant. The second limitation is, of course, because you train that generative model on multiple sequence alignment, if that MSA is not very deep, you may run into trouble training your model. You may not have enough data to learn a very good generative model of that protein family. And so that led to this other stream of ideas of learning a generative model across protein family with language models, so initially with RNN-based approaches like UniRep, and then progressively mimicking a bit the trends that were happening in NLP, various transformer-based architectures, to learn a good generative model of protein sequences across families. There again, it was I think very promising to learn this general purpose representation, but when looking at the performance of those models, they were not performing as well as the alignment-based counterpart. Right? Like so, even like the like the ESM and ProGen models, without any fine-tuning, for instance, the performance was not matching the best model classes on the alignment-based strategies. And so that led to various strategies to combine those two ideas. So combine the strength of the alignment-based models and combine the strength of those models that are family agnostic. And if we step back, there's like three broad strategies that have been proposed to do this. The first one is you can fine-tune your PLM, your protein language model, on a set of homologous sequences. So, for instance, you fine-tune your model on an MSA, and that leads to good results, but it's sometimes a bit finicky to get it to work. Another very powerful idea was to do retrieval at training time. So you learn a distribution across multiple sequence alignments. And so an example of this is MSA Transformer. A more recent example of this is POET. But like the goal is to you collect sets of protein sequences that belong to the same family, and then you learn a generative model over those protein sequences. And the last idea, which is what I'm going to be focusing on in the next few minutes, is retrieval at inference. And I'll describe this in the next few slides. The first architecture that developed this idea is transception. In transception, you have like two core contributions. The first one is this transception self-attention mechanism, which is interesting in its own right, regardless of retrieval. And the idea there was to have an inductive bias that promotes in the architecture capturing information at the levels of k-nodes that we know are important in protein modeling. And what we wanted to do is to split the different attention heads and then apply different convolution kernels of different sizes on each of those attention heads, so that each head specializes to the extraction of patterns at different resolutions. And that worked very well in practice for protein modeling, much better than the vanilla GPT architecture. The second contribution of that paper was to combine those two modes of inference, so the autoregressive inference that you get from a bona fide autoregressive transformer, and then this retrieval at inference, which effectively what we do is we retrieve a multiple sequence alignment for the protein that we care about, that we want to score, and we look at the distribution of amino acids at that position to complement the autoregressive transformer will make a prediction at this residue based on this left context. And then what the retrieval at inference time will do is to complement that based on the distribution of amino acids that have been seen in homologous sequences at that position. And combining those two led to very strong performance, in particular when the MSA is of very low depth compared to the traditional alignment-based models. Building on this idea, or one reason that I think retrieval at inference is quite compelling is that gives you a lot of flexibility to craft the multiple sequence alignment to the particular application. And so if you have particular domain knowledge for that protein, so for instance, in the case of BRCA1, we know that there are two very clearly identified domains for which it is very easy, or like it's much easier to get good quality MSAs for each domain separately. Because you have the flexibility to do that as you want at inference time, that makes this type of incorporating this type of information much easier. Another idea that we decided to build upon in that work was, instead of looking at the distribution, like making that prediction based on the distribution here of amino acid at that single position, we decided to leverage the constraints across the full sequence for the retrieved sequences in the MSA. And that led to transceptive, which is pretty much the same idea as transception, except that we recover an Eve model and predict the log probability at that position given that alignment-based model. And that leads to very strong performance in practice, pretty much like on the protein gene benchmark, reaching good performance in pretty much all the different settings, and also for clinical annotations. So those ideas are very powerful. I encourage you to think about how you may want to combine sequence-only information with MSAs in your own pipeline. I think it works. You need to do that for fitness prediction, and as you may know, it's also important for structure prediction as well to reach state-of-the-art performance. So that's, I think, a general idea in protein modeling. So are we done with those sequence-based models? So the answer is no. There's plenty of things that we can do beyond the architectures that I was just presenting. And just to illustrate the idea, this is a diagram that shows the performance of the best zero-shot approach on the benchmark. So transceptive versus the best supervised approach in blue, and so that's a model I'm going to be talking about in a minute. But that shows that if you do have particular information from your experiment, there's a lot of value in leveraging that to your application, and there are various cases where you have to do this, and relying solely on evolutionary sequences is not going to do the job. So how it's been done in the past was primarily based on those discriminative models that leverage various ways to represent the protein sequence, so either one-hot encoded representation or physicochemical property features, or more recently embeddings of those protein sequences that are obtained from a protein language model, and then trained on on property data that you collect from experiments, but because you don't have a lot of those data points, the classification head or the regressor head was typically lightweight to not lead to overfitting. And what has been suggested a few years ago was to augment those discriminated models with zero-shot fitness prediction, so with the types of models that I was talking about in the first half of this talk, and that's work that has been proposed by Chloe, Sue, and colleagues, and that leads to very good performance. So that's something that is useful to do in practice. What we wanted to do with protein NPT was to do, well, several things. So we wanted to learn a strong generative model of both sequence and labels, and that's I guess the rationale for doing this was to then be able to condition on particular label values to then sample from the sequence part for sequences that would be more likely to have the properties that you care about. So what we do here on, I guess the right way to, or like the simplest way to describe what's going on this figure is to start from the bottom left. And so we jointly embed in the same space protein sequences with target values, right? And then we mask a proportion of those tokens in the sequences, and we mask a proportion of targets in the target feature. Then we apply several layers, protein NPT layers, and I'll describe that in a second. And then the signal that you use for training is this hybrid loss of mass token reconstruction and target prediction. And what happens in the protein NPT layer is what we do is it's an idea that stemmed from actual attention that was used in computer vision initially, where you're going to reduce the complexity of performing self-attention across your entire two-dimensional tensor by successively applying attention across rows and across columns. Actually, first across columns. So for a given sequence, you're going to do self-attention across your different amino acid tokens and labels. And that is typically if you use an ESM model, for instance, that's the type of attention that you typically perform minus the label. But like trying to learn a representation of like the dependencies between residues and labels. And then you do column attention. So a particular residue, you're going to do self-attention across rows to learn homology between those different label sequences. So that's, and the last piece is the standard feedforward layer. So that's what's happening at train time. At inference, it's pretty much the same thing, except that you do not mask anything on the sequence side, but some of the targets are masked by default. But these are the targets that you want to predict. And the value of it, I think, is you get those two modes of inference. You can predict a missing target value based on the dependency between the amino acid residues and the target, but also leveraging the column-wise attention based on the homology between different labeled sequences. You're going to look at the similarity between the test sequence and a sequence from your training data, and you can also get a prediction that way. That works very well in practice on the various protein gene benchmark tasks, both for single and multiple substitutions, but also it's very easy to predict multiple properties at the same time. In that construct, you would just need to add one column for each target that you care about. And it's very useful for protein design. So what we tested was iterative optimization of those protein sequences in a Bayesian optimization setting. And so we introduced various measure of uncertainty prediction with that architecture that can be used in the standard Bayesian optimization acquisition function. I wanted to conclude with that analysis of conditional sampling. So because you've learned that generative model of sequence and label, now you can condition on a particular, let's say that you wanted to redesign an enzyme to be more thermostable. You would condition your model on very high value of thermostability and ask the model, mask a few positions in your sequence and ask the model to predict sensible values of those amino acids given the target that you hold fixed at a high value. And that has proven very effective in experiments. Look, we've focused on in silico experiments so far. We are testing it in real wet lab experiments at the moment. But that's very promising, and I think intuitively that's something that is quite exciting to go after. So I'll conclude with this. So we've seen that to reach state-of-the-art performance for fitness prediction, we need to leverage hybrid models that combine alignment-based strategies with models that are trained across protein families, so protein language models trained across protein families. We've seen that we can use sequence label models to further boost the performance of zero-shot prediction. And then more generally, those different approaches can be quite powerful to optimize proteins even in non-natural conditions. There's lots of, going back to the slide that I presented earlier in the talk, there's lots of very exciting work that happened at the boundaries between those. So I presented those models as different archetypes that seem isolated on this page, but actually the frontiers between those different modeling strategies are becoming, or they are fading away, if you will. And so I've highlighted a few of those more recent works that combine sequence and structure for protein modeling that you might be interested in. And I conclude by thanking all of the amazing collaborators that had the pleasure to work with over the past 2 years on these various projects. Thank you very much for your attention, and I'll take any questions.
Amazing, Pascal. Thank you so much for bringing this all together for us. We have a few minutes, I guess, for questions. So, are there any questions in the room already? I think we've got time for like a couple of questions at most. No, I have a few. One thing you told us about in the start is that basically you see these 3 different pillars for protein modeling, right? Sequence, structure, potentially looking at evolutionary information as well. And making a little bit of a connection, I guess, with Lucy's talk, who introduced quite interestingly the language idea. Are you thinking about also incorporating other more like unstructured information? Long been following this domain, I've always thought, isn't there context about proteins that would be valuable to integrate in some of these predictions?
Yeah, that's a great point. So I said like that's 3 core modalities that I think for protein modeling. There are like many more that are not on this page. I think like incorporating language information is a good way to perhaps go around some of the limitations that we have from the third bucket. So by sifting through various papers and finding a way to bake that into the model somehow. I think it's still very early stage for those models, but it's very promising to see those papers coming up. I think they will ultimately provide a more intuitive interface between those protein modeling models and the end user. I think it's still early days for those, but super exciting work.
Amazing. Thank you so much, Pascal. I think, are there any further questions in the room? One question here.
I was just wondering if you tried this out for antibodies, or give an idea if it would work well for that, when you've got a lot of diversity within a single class.
So the question was a bit hard to understand over here.
I was wondering if you use this for antibodies or investigated whether a kind of data retrieval would work well for antibodies.
Yes, and is your question specifically for like in that first bucket of sequence-based models? So like, I guess you could.
How using the data retrieval specifically?
Yeah, so like I've not used that for antibodies specifically, but I think the more generally those approaches you can train those models on the entire set of protein families that you typically have in UNRF, etc., and Magnify and BFD, etc. Or you can specialize them on, let's say, antibody sequences or enzyme sequences, and the same types of approaches would work. There's no theoretical limitation, at least. I've not tried it in particular on antibody sequences, but I think it would be promising to give it a shot.
I mean, I was just wondering, because you have such a high rate of mutation in antibody sequences compared to other protein classes, if that would be an issue, or kind of the limits on how much data you could include with this data retrieval.
So like it would be interesting to do some various experiments there. I think like you would have to perhaps innovate a bit to go around the limitations for antibody sequences. I think so. You can either like downsample the retrieved sequences, or there's always a bit of that dark magic at play whenever you create a multiple sequence alignment, even for enzymes, and that's relying on heuristics from the field to select the homologues that are the most adapted to your particular application. And so there will be a bit of a tinkering there to get it to work well.
Amazing, cool. Pascal, thank you so much for coming to us today and jumping in on short notice here to help us out. Really, really amazing to hear all of this, and appreciate the session that you gave us today. Thank you, Pascal. Cool, with this we're moving on to our last but not least speaker of today, David Baker. David really needs no introduction, I believe, but he's really the pioneer in protein design. Rosetta is a technology that he developed, as well as protein structure prediction. So it's really a pleasure to introduce him to this forum today. And David, please feel free to take it away.
Let's see. Sorry, just trying to find my slides, and I'm a little disconcerted by the two pictures of me on the screen. Let's see. Sorry. Here we go. Let's see. Is that working okay? Can you see my slides?
Yep, we can see it clearly.
Okay. All right. So I'm going to thank you very much for the invitation, and I really enjoyed the talk. Let's see. So I'm going to talk about de novo protein design. I think probably what's different about my talk from the other talks is that I'll show you a lot of experimental data. So we're constantly going back and forth between kind of developing new methods and then testing them in the lab. And so I'll tell you a little bit about the new methods, and then a lot about sort of the kinds of things we can do with them for protein design. So before I get to design, we've been working for a long time on protein structure prediction, and this is now published, but just wanted to briefly mention this, that with AlphaFold and with RosettaFold, you could predict the structures of proteins pretty well, but generating modeling of all the components of a biomolecular system was hard. And we've now generalized RosettaFold to what we call RosettaFold all-atom, which really takes in inputs of basically all the different components of a biomolecular system. Proteins and nucleic acids are represented through their sequences, and everything else is represented through an atom graph, and that lets us model all kinds of different structures. And then we can also, since RF diffusion is based on RosettaFold, this now enables us to design proteins that bind to small molecules and catalysts by carrying out diffusion around the small molecule. So this is just what I said before. We represent the the sequences, the polymers, the sequences, the other molecules, components as atoms, and we have an atom graph to describe their connectivity. I think this one takes a little while to play, but anyway, so we can now generate models of proteins around small molecules, and this isn't as accurate as we would like it to be. We're working to improve the accuracy, but we can get accurate predictions for compounds that are quite dissimilar to the training set. So it's not just memorizing. We can also model covalent modifications on amino acids and metals and so forth. But most of what I wanted to talk about today is protein design, and so we generally approach design problems with first defining what the problem is, then running RF diffusion to generate a structure which is sort of conditioned on whatever the problem is, then protein MPNN to design a sequence which will encode that structure and the interactions that the protein makes with the target. So we recently generalized this to be able to model proteins in the context of DNA and small molecules. We call that ligand MPNN. And then finally, we use AlphaFold or RosettaFold to predict the structure. And now, as with RosettaFold capable of modeling small molecules, we can test protein small molecule designs by holding the sequence and the small molecule together, just as I just showed you. So as you know, diffusion models are based on sort of iterative denoising. So this just shows an example from images. I think you're all familiar with this. In the case of RosettaFold RF diffusion, we start with native structures. We noise them to different extents. We fine-tune RosettaFold to remove the noise, and then we can start with completely random distributions of amino acids and progressively denoise to get what are very plausible protein structures. And so this is really working quite well. So here's a nice example. So this is the TNF receptor, which is a long-standing drug target because it plays a key role in autoimmune disease. So there's a lot of antibodies that have tried to target this. And so here you see, just starting with the completely random distribution right there around the TNF receptor, and diffusion generates this really pretty incredibly shape-matched structure, which kind of cradles it. And you can see the details of the interactions here. This binds quite tightly already, and then basically we can take the best few things we find in the first round. We can add noise again, a smaller amount of noise, and then remove the noise, call this partial diffusion, and we can get down to single-digit picomolar. And since these designs have such an extensive surface of interaction and are associated complementary to the target, we can then adapt them to other family members, and many of these are therapeutic targets just by carrying out this partial diffusion and then denoising in the presence of different targets. So we've been able to pretty systematically go through and make high affinity and specificity binders to these targets. It's kind of exciting for us because we tried actually with our previous Rosetta kind of physically based methods quite a bit to make binders to this target. It was very hard because it was flat and pretty polar. So we've generalized these methods to nanobody and antibody design. So here again, we start with the random distribution residues, and then the denoising generates a nanobody in this case. And here's the cryo-M structure of this nanobody bound to the target, and you can see the loops are in pretty much the right place, and the overall structure is in the right place, and the binding mode is correct too. So since all we're doing is we're specifying where the region is, but not really how it's bound or what the loops look like. So we're pretty excited about this. I just threw this in here just to say that we can use these approaches to make proteins that have kind of remarkable, very promising effects in animal models. So this is a design that mimics the IL-21 cytokine, and our collaborators, Stephanie and Michael Dugan, are doing a variety of animal tumor model experiments, and this compound is really looking extremely good in extending life, even in difficult models like pancreatic cancer. So we're quite excited about the possibilities. So that was sort of design of binders to folded targets, but we can apply these methods to disordered regions too. So for example, we can carry out the diffusion around peptides. We can either keep the peptide fixed, or as in this case, or we can allow the peptide to move at the same time. And these binders that come out of this can be very high affinity, with picomolar affinity, and we've solved structures of a few of these, and they're quite accurate. We've been particularly interested in peptides that form amyloid, obviously for trying to contribute to the treatment of amyloid disease. And so in this case, we assume that the peptide samples some fraction of the time in extended chain conformation. And if it does, we can, so we can design binders that will sort of bind it from both sides when it's there. And these binders can bind pretty tightly. And here is a binder to A beta. You can see this is work, collaboration with Thomas Knowles' lab in Cambridge. So when they add A beta or tau to a test tube, it aggregates. And as they add increasing amounts of these design proteins, it suppresses aggregation. And it's kind of neat. They suppress aggregation even at slightly substoichiometric amounts. So they seem to be interacting with the fiber as well. So we're excited about applications of these to, like I said, to diagnosis and treatment of neurodegenerative disease. And more generally, there are a lot of proteins in the cell which are disordered, and it's been very hard to make antibodies against them or detect them. But we can use the same kind of method to design binders which bind to these regions. This is work by Keija Wu. And when we make these binders, we find that and put them in cells, we can specifically recognize not only the target protein, but sort of pull down all the other proteins that they're interacting with. So that's pretty useful. So we've also been very interested in working on in designing transmembrane proteins, and in collaboration with Anastasia Vorogava's group in Belgium, we've been designing nanopores that these are beta-barrel nanopores that insert into the membrane, and they have these pores of different sizes. And as the pores get bigger, the conductance, we can measure the amount of current going through the pores, increases as well. And what's neat is we can sort of put together different modalities. So as I briefly mentioned, we've been designing proteins to bind to small molecules. This is a binder to cholic acid, and here we have a crystal structure as shown here. So the design protein is really making extensive interactions with the cholic acid. What we can do is then sort of this design is, we call it a pseudocycle. You can see it as rough, approximate threefold symmetry. So we can take each of these helical hairpins and attach it to one of the hairpins on one of these nanopores, and to make something that looks like this. And then it's kind of neat when you measure the conductance without cholic acid, we have sort of these transient closing things, steps which we think are the binder coming together without the small molecule. But then when the cholic acid is added, the conductance changes overall, and we get these long, completely closed periods. So we're very excited now about making sort of a general molecular nose where we make a bunch of these pores, and what we find is that different small molecules affect different pores in different ways. So kind of like in the way our noses work, where we can distinguish, or at least dogs can distinguish, a very large number of different molecules. So this is kind of a neat problem, and it is also going to be a fun one from the ML point of view in the deconvolution step. So now I'm going to switch gears and talk a little bit about protein nanomaterials. So it's an exciting time. We have our first de novo design medicine that's approved for use in humans. That's work from my colleague Neil King, a COVID vaccine based on these self-assembling icosahedral nanoparticles. And so what we've been looking at more recently is, well, first of all, sort of Helen Eisenach has been been sort of extending the diffusion to icosahedral structures, in particular ones where we can specify the geometry of for example, this trimeric unit. And that's important because if we want to scaffold a virus, in this case, respiratory syncytial virus, glycoprotein, we really need to have the geometry right. And this is a cryo-EM structure, so it just carries out the diffusion purely specifying the geometry of the sort of internal helices here. She gets these very beautiful cages that match pretty closely to the design model. But what we've been sort of focusing on is the problem of how to break symmetry. Because if you have pure hexagons, you will get a hexagonal lattice. I think everyone's familiar with that. And if you have pentagons only, you get an icosahedron or a dodecahedron. And if you want to go in between, you have to break symmetry somehow. And so there are two ways we've been exploring that. One is with pseudosymmetry, where we have two types of components. One that's forming, and the two types basically let us break symmetry. So here we have the hexagon and the pentagon. You can see they're surrounded by different collections of these molecules. The other way we can do it is make somewhat flexible trimeric components that can then adapt to their environment to form either pentagons or hexagons as needed to reduce strain. We call the first one pseudosymmetry and the second one quasi-symmetry. So this is an example of pseudosymmetry where here's an icosahedron that's been designed, and this is a cryo-EM structure, with two different types of subunits. One and that allows us to break symmetry, as I just showed. Here's the hexagons and here's the pentagons with the different types of trimers shown. And for reference, this is the size of AAV, which is a current gene delivery vector. So we're excited about being able to make things that can package more cargo. For the second step, quasi-symmetry, we need to interpolate between the hexagonal case and the purely pentagonal case. And we can sort of parameterize this system, the angle between the arms of the trimers, and then the degree to which they depart from being perfectly flat, as shown here. And as we move along this curve, we find we generate different structures. And so, for example, in this region here, when we design trimers with this property, we get structures that have that single protein component, but symmetry is broken because here's a hexagon, here's a pentagon. And we have the cryo-EM structure now, so we can see exactly how this happens. And then for those of you who think protein structures are a real pain to look at and way too complicated, we've been designing proteins that are much, much simpler, and where protein design becomes something like putting together, wooden pieces of wood in a lumber yard. So we made these like curved proteins, these straight proteins, and we can put them together in different ways to make objects like this, like these. And these are EM structures. And then if we want to change the size, we just change the size of this linear block here to make things that are progressively larger. So that really, I think, can simplify protein engineering. We can also make nanomaterials that switch states. So here is a tetrameric ring that Arvind Pillai designed, and when he adds an effector, it switches to a pentameric ring shown here, and you can see the change in the EM here and in the mass here. So we can now design systems proteins that fold not just to one state, but to two states, and where we can toggle that by adding an effector. And what we're trying to do now is to make this driven by a chemical reaction, so the system will move back and forth between different states to make molecular motors. And then finally, I want to say a little bit about DNA binding proteins. So we've been designing small proteins that bind to DNA. Here's the designed protein, and here's the bases that are being contacted. And they're quite specific. So if you change the amino acid, change the bases that in the recognition site, you no longer get binding. And so we've actually been able to make now binders to quite a number of different DNA sequences, and we can put them together, these binders together, using diffusion. So these are the original binding domains, and then we're diffusing these linkers between them and control the spacing, and we can get repression in bacteria and activation in mammalian cells of DNA sites which match the spacing and the recognition properties of these binding domains. And so we're now generalizing diffusion to both diffusion in the presence of DNA, which is how we're now designing these DNA binding proteins. I mentioned we have ligand MPNN, which we now use for designing the interface. And we're also using this to design RNA binding proteins, as illustrated here. And finally, I just want to show you that these methods can be applied beyond the peptide bond. So this is work from Patrick Salveson. So we were interested in incorporating chemistries that go beyond the peptide bond that's found in proteins, and he's been able to design a variety of small molecules very precisely that adopt unique shapes and have distinctly non-protein-like chemistry. So that's kind of an overview of the various things that we're excited about these days, and I think collectively it's a really an exciting time for protein design. I think there's a lot of applications in medicine, in technology and sustainability, and this is just a short list, so I'm really excited that more people are getting interested in this. I think there's a ton to do. And let me just briefly acknowledge the people who did this. Min-Kyung, Frank developed the original version of Rosetta Fold. Frank, Ju, and Rohith, particularly Rohith, have been the drivers behind Rosetta Fold All Atom. RF diffusion, work of Joe and David, many people, outstanding people listed here. I mentioned Helen extended this to symmetric assemblies. Woody extended it to small diffusion around small molecules, and Preetham and Susanna did the peptide diffusion work. The protein binding has involved many great people over the years. I showed you Danny and Hannah's work on the amyloid peptide binders, Kaja's work on the recognition of proteins inside cells. I didn't talk about these things. For DNA binding, Cameron, Robert, and Ryan developed those specific binders, and Paul Kim has been extending this to diffusion. And for RNA and protein RNA diffusion, Andrew and Pam have been working on this. I showed you Sang-Min's quasi-symmetric and pseudo-symmetric cages. The nanopore sensor work is done by Samuel Saigorev and with Carolyn in Anastasia Vorobyeva's lab. And I showed you Arvind Pillai's sort of two-state systems that were switching from one state to another. And I've had really outstanding collaborators at the Institute for Protein Design. So I'll stop there and be happy to take any questions.
Thank you so much, David. Really exciting tour through the world of protein design that you gave us today. As always, we have a few minutes for questions in case somebody has something. There's a hand raised here. Go ahead.
Hi. Really great talk. I'm not super familiar with RF diffusion, so this question might be a little bit naive. But I was wondering if it is possible or feasible to use like RF diffusion to design proteins with chemical modifications. For example, if it is possible to design some kind of glycosylated protein with glycosylation patterns being restricted to a certain type of branching patterns, or to be composed of certain sugar moieties. Thank you.
Yeah, so with Rosetta Fold All Atom, I mentioned that we've now extended RF diffusion to being able to model, sorry, I have to not look at myself. That probably doesn't work either, does it? Okay, yeah, so we can now design proteins to bind to, for example, covalently modified proteins with containing sugar groups, and we can also design proteins that wrap around, that contain glycosylation sites that wrap around them to try and control what the sugars are, how the carbohydrate is processed. So I don't think we've done exactly what you described, but I think that's certainly possible now with the latest version of RF diffusion.
Thank you.
Very cool. There's another question here in the back. Maybe come a bit from easier to hear.
Hi, can you hear me?
Yeah, it's okay.
Can you hear me? So thanks so much for the talk. I had a question about some of the limitations of the publicly available data, especially with respect to structure and things like RF diffusion. Is paratope, epitope information essential to these types of models? And with respect to immune receptors, antibodies, things like this, how do you view the flexible loops?
Right. So, the training data is pretty much just the structures, right? And certainly we are limited by the number of structures in the PDB. Particularly, we're doing a lot of work on T-cell receptor peptide MHC recognition, and there's not as much data as we'd like. Certainly more antibody data would be good. Also more small molecule complexes. For the loops, for example, that the RF antibody or the RF diffusion for antibodies, they are just getting built from scratch. So we give it a framework, something that expresses well, and then we specify the target and the site, and the loops just basically come from scratch along with the binding mode. So that's essentially the problem that the diffusion is describing, is how to place the framework and how to model the loops to get highest affinity binding to the target.
Thank you. Cool. There's another question in front.
This might be a bit crazy, but so have you considered trying to design a protein to work as a semiconductor or a transistor?
Yeah, well, there are kind of two ways to think about that. One is you can develop, you can design sort of like protein circuits where they're transistor-like proteins and proteins which modulate their activity. And we've actually been doing that. So we have proteins now which we design a system where we have allosteric modulation. So that means you have a protein which does something, and then you have another protein which binds at a distance site which modulates that. So for example, we've designed proteins that bind to the IL-2 receptor, and then another protein which will bind to that protein and make it come off. But the other way to think about your question is to actually modify silicon, right? To basically have proteins that modify existing sort of transistor-like geometries. And we haven't done exactly that, but we're doing quite a bit now on at the interface between proteins and silicon. So for example, we've been able to design pores that will insert into pores, into nanopores in tried chips. And that's a very exciting juxtaposition, because then you can potentially get the advantages of recognition of biology with all the convenience and electric circuitry of silicon. So I think both answers to your question, both aspects of your question are very interesting. Both sort of synthetic protein systems that behave like electronic systems, but purely in the protein realm, as well as hybrid protein silicon systems.
Very cool. There's another question here.
Yeah, a little bit of a follow-up. Thank you for the talk. I've been told DNA, the de novo synthesis is quite of a problem if you don't have a template. Have you thought about designing proteins like nanopores that, given a current, add an ATCMG in a consecutive manner that de novo synthesis of DNA can be much faster?
Well, that would be neat. I mean, so, there's been a lot of work on DNA sequencing, sort of the opposite of what you've described, and we're trying that out with our protein silicon systems. I think as far as sort of things, there's enzymatic systems like terminal transferase-based systems, which add on bases. But I think there's a lot of different ways to approach that problem, and I think it is a very important one. We're not specifically working on the synthesis problem, but I think that is a very interesting application of these methods.
Very cool. Another question here.
Yeah, are you restricted to naturally occurring amino acids, or can you go for more synthetic amino acids and do even more beneficial proteins?
Right. So I showed at the very end that we can go beyond the even natural protein backbones, done natural backbones. it sort of, we have a combination of the physically based methods, the Rosetta methods, and the deep learning methods. So the Rosetta methods, you can put in anything you want, unnatural amino acids, unnatural backbones. You have to play tricks with the deep learning methods because what your database is all L amino acid with the 20 amino acids. But there are various ways you can do this. For example, you can invert the PDB and then get a very good training set for D amino acid protein. So we're basically doing a variety of those things and combining with the physical methods to be able to model unnatural amino acids. We can also atomize in both Rosetta fold all atom and the ligand MPNN. We can atomize targets or parts of a protein, and once we've atomized it, it can be represented, it can really be anything. It doesn't have to be a natural amino acid.
Makes complete sense. With that, I think we have covered all the questions in the room. David, I think I speak for the whole room. We really enjoyed the session and really appreciate the time that you've given us out of your Monday morning here. And thank you so much for giving us the session.
Okay, thank you.
It's a great honor to give you the last words on this session here today. I think just to go back to the founding principles of why we started the MLDD, it was really supposed to be a place to bring together the community to really share knowledge and exchange information. And hopefully, spark a little bit of the inspiration that gets us back to work. And as you all know, I think drug discovery and machine learning drug discovery in specific, I think it's now the second most interesting investment area for a lot of people. And so that comes with a lot of responsibility for us to really make the most out of that responsibility investment that is coming to us. And so we want to really make the highest chance that we actually reach patients with our medicines, with these ideas and these potentially new cool tools. And so I want to thank you all for being part of that and coming here to join us for this session, really trying to live the spirit of that. And also a big thank you to obviously the whole team here supporting the sessions and the whole organizing group as well, making this happen. Thank you so much.